Welcome to the workshop today entitled Mortality and Antipsychotic Use in Dementia-Related Behavioral Disorders. My name is Nancy Allen Lapointe, and I'm Adjunct Associate Professor at the Duke School of Medicine and a Faculty Fellow at the Duke-Margolis Institute for Health Policy. This workshop is being convened by the Duke-Margolis Institute and our partners at the U.S. Food and Drug Administration. During this virtual workshop, you'll hear insights from experts on a reanalysis of the data that supported the regulatory action related to the boxed warning for antipsychotics used in adults with dementia, as well as findings from a recently conducted literature review on this topic. You'll also hear reflections from participants on the available evidence and further considerations related to the assessment of risk associated with the use of antipsychotics within this patient population. During the 4 scheduled sessions of the workshop, we will hear presentations and moderated discussions exploring these topics. Before we begin, I'd like to go over a few considerations for our discussion, as well as some housekeeping notes. First up on the screen is our statement of independence. We'd like to acknowledge our commitment to academic independence and to avoid taking partisan positions in our work. You can find more information about our policies at our website and via the links on the slides. Next, here are a few reminders about our meeting format that will help us organize discussion in this virtual setting. A reminder that the meeting materials are available on the Duke-Margolis website. To our attendees, if you would like to ask a question during the presentations and open discussion portions of the meeting, please type it into the Zoom Q&A box. Please be aware that due to the high volume of meeting participants, we may not be able to get to your question, but we'll try to get to as many as possible. A recording of the meeting will be available on the Duke-Margolis event website and YouTube channel after the conclusion of the event. Today's slides will also be made available on our website in the coming days. Finally, the Duke-Margolis Institute is convening today's meeting in collaboration with the FDA. This is not a federal advisory committee meeting. While we are keenly interested in your input on this topic, we won't be following advisory committee procedures or taking a vote at the end of the meeting, etc. The meeting will be a success if there is an exchange of lots of ideas and open discussion. We are thrilled that you are all able to join us today, and we anticipate an interesting and productive discussion. Before we jump into the meeting, I'd like to go over today's agenda. After the opening remarks from the FDA, we're going to begin today's workshop with 2 presentation-focused sessions. The first session will include an introduction to antipsychotic use in dementia-related behavioral disorders, and the second discussion will feature a review of data analysis and literature review findings. Each of these sessions will include some brief time for speakers to also answer clarifying questions from the audience. After a short break, we'll return for a discussion-based session of stakeholder perspectives on considerations regarding the use of box warnings for antipsychotics. And then our final session of the day will have a panel discussion on opportunities for further characterizing value and need for box warnings for antipsychotics. Finally, we'll have some brief closing remarks and then adjourn today's event at 4 p.m. Eastern Time. Now, I would like to pass things along to Dr. Tiffany Farchione. She is the director of the Division of Psychiatry at the FDA, and she's going to share opening remarks from the agency. Dr. Farchione, take it away.
Great, thank you. So as was already mentioned, my name is Tiffany Farchione. I'm the director of the Division of Psychiatry at the Center for Drug Evaluation and Research at the FDA. So we are the folks who deal with psychiatric drug indications, not devices or biologics. So in any case, I just want to start off first by thanking the folks from Duke-Margolis for helping us put this workshop together, and also for the folks that you're going to hear from today in their various presentations. I think that the audience will quickly realize that a lot of work has gone into preparation for this workshop, into the data analyses and everything behind the scenes. So I just wanted to start off by making those comments and thanking the teams involved. So if we go to the next slide. So just to give us some context for why we're here today, Alzheimer's is obviously, it's the most common cause of dementia, and we have about 6.5 million people in the US diagnosed with Alzheimer's disease over the age of 65. And even though cognitive decline is the predominant symptom that you see in dementia, neuropsychiatric symptoms are common. And this is a heterogeneous range of non-cognitive symptoms that can include agitation, aggression, delusions, hallucinations, as well as things like depression, anxiety, apathy, disinhibition, sleep disturbances. It is a very broad category that we're dealing with when we talk about neuropsychiatric symptoms. You'll also sometimes hear this referred to as behavioral and psychological symptoms of dementia, or BPSD, but for the purposes of today's discussion, we're using the neuropsychiatric symptoms NPS designation for these issues. NPS are an important focus of attention clinically because they're associated with a higher risk of accelerated disease progression, with functional decline, with worse quality of life and greater caregiver burden, as well as increased out-of-home placement and earlier death for these folks. And the clinical presentation and frequency of neuropsychiatric symptoms can vary, but most patients will experience an initial onset of symptoms in the later stages of dementia and worsening symptoms as dementia progresses. Next slide. And this is, of course, an unmet medical need. The clinical management of neuropsychiatric symptoms is a challenge. Non-pharmacological approaches are typically considered first line, and these include things like cognitive stimulation, group therapy, exercise, music therapy, multisensory therapy. And then there are off-label pharmacologic options like benzodiazepines, antihistamines, antidepressants, antiepileptics, and most antipsychotics. But the studies that evaluate off-label pharmacologic treatments are really also very heterogeneous in design and in the patient population. And the results of those studies have really demonstrated only small improvements in efficacy, but with serious risk and tolerability concerns. Next slide. So in terms of the use of antipsychotics for the treatment of neuropsychiatric symptoms, as folks are aware, and part of the reason why we're here today is that in 2005, the FDA added a boxed warning to all of the antipsychotics for the increased risk of mortality in elderly patients with dementia-related psychosis who were receiving antipsychotic treatment. And after the implementation of the box warning, various regulatory bodies and healthcare institutions have taken action to decrease the off-label use of antipsychotic prescribing. But drug utilization data suggests that there's been a decrease in antipsychotic use, but a concomitant increase in the use of things like opioids, antiepileptics, benzodiazepines, et cetera, among elderly patients with dementia. Next slide. So antipsychotics are currently used as a first-line treatment choice. The American Psychiatric Association practice guideline recommends the use of non-emergency antipsychotic medications for the treatment of agitation in patients with dementia. We do have one antipsychotic approved for agitation in Alzheimer's disease, and we have one for hallucinations and delusions in Parkinson's disease, which does also include Parkinson's dementia. But other antipsychotics are commonly prescribed off-label, despite the limited benefit described in the current literature and the increase in risk of mortality as outlined in our box warning. So next slide. So, in terms of the focus of the workshop today, again, as I noted, we had the box warning in 2005 on all the atypical antipsychotic medications based on the analysis of randomized controlled studies that were available at that time that showed an increased risk of mortality in older adults with dementia-related psychosis. But over time, obviously we're going on 20 years since the box warning was issued. We have additional scientific evidence that's become available, clinical practice guideline changes occurring since that time, as well as the introduction of new treatments to the market. So that gives us an opportunity at this point to reevaluate the need and the value of the box warning that's included in the approved labeling of antipsychotic medications for these impacted populations. Next slide. So this workshop is going to be the first step that we're taking to initiate a dialogue with the scientific and clinical community, with patient representatives, with other stakeholders. What we're presenting today is not a final assessment. This is the first stages of this process for us. So folks who are here today are going to hear insights from experts on a reanalysis of the data that supported the regulatory action related to the box warning, as well as findings from a recently conducted literature review. And the attendees will also hear reflections from the participants in the workshop on the available evidence and further considerations related to the assessment of risk associated with use of antipsychotics in this population. But I do want to emphasize that the workshop isn't going to be focused on methodology issues related to the development of treatments for neuropsychiatric symptoms. This isn't going to be about efficacy. What we're really focused on today is going to be about the evaluation of safety in these populations. So if we can move ahead. That pretty much covers it for our introduction, and I will pass it back to Duke-Margolis. Thank you.
Thank you so much, Tiffany, for those opening remarks and really doing a great job of setting the stage for today's workshop. And I think this first session is going to kind of continue to set the stage. So we are going to transition to that first session that's titled Introduction to Antipsychotic Use in Dementia-Related Behavioral Disorders. During this session, we're going to hear from two presenters that will provide the background and context, or further background and context for this workshop. As a reminder to the audience, you can submit your questions through the Zoom Q&A function throughout the session, and we will have time at the end for a few clarifying questions on the content that was presented. We'll do our best to get as many of those in as possible. And with that, I'd like to introduce our first speaker, Julia Bernot, a neurologist serving as a clinical reviewer in the Division of Neurology 1 in the Center for Drug Evaluation and Research at the FDA. Julie, I'll turn it over to you.
Thank you very much. Hi, thank you so much. Good afternoon. I will be presenting a brief background of neuropsychiatric symptoms of dementia to lay the groundwork for upcoming presentations. Next slide, please. The general term dementia has been evolving. Dementia may be caused by various neurodegenerative disorders, the most common of which is Alzheimer's disease. Recent advances in vivo biomarkers have allowed for a biological definition of Alzheimer's disease rather than a syndromic one. The randomized controlled trials that were reviewed for the purpose of estimating the risk of mortality with the use of antipsychotics as treatments for behavioral symptoms of dementia, which is at the core of today's discussion, utilized clinical definition of Alzheimer's disease and certain related dementias. Without the ability to differentiate dementia etiologies biologically, it's not possible to determine which type of dementia affected the patients enrolled in the studies. Next slide, please. Neuropsychiatric symptoms, as was already stated, also commonly referred to as behavioral and psychological symptoms of dementia, are a heterogeneous group of non-cognitive symptoms that include disturbances of mood, behavior, thought content, and perception. They are experienced by over 90% of patients with dementia of any cause during the disease course. They are also commonly exhibited by individuals with mild cognitive impairment, that is, cognitive impairment not severe enough to affect daily life in the pre-dementia stage. They may even precede cognitive impairment. The etiology of the symptoms is complex and includes biological, psychosocial, and environmental factors. The frequency of the symptoms typically increases with the severity of cognitive decline. Symptoms may persist, remit, or be episodic over time, and this varies between symptoms. Next slide, please. Certain neuropsychiatric symptoms co-occur. For example, anxiety and depression are frequently present together in individuals in earlier stages of Alzheimer's disease. Some symptoms or clusters of symptoms are more likely to be exhibited in certain neurodegenerative disorders. For example, hallucinations, particularly visual hallucinations, or more common Lewy body disease than in Alzheimer's disease. A wide range of neuropsychiatric symptoms is common across dementia types. The symptoms listed in this slide are not exhaustive. Many studies have attempted to identify sub-syndromes of neuropsychiatric symptoms through factor analysis of cross-sectional data. The purpose of the analyses included finding a common neurobiological pathogenesis of symptoms and to inform medical management. One of many ways of grouping symptoms is shown here, but no consistent method has been identified. Next slide, please. Since the initial box warning of antipsychotic medications in 2005, expert consensus diagnostic criteria have been developed for apathy, agitation, and psychosis, which are among the most common symptoms in dementia of any cause. And importantly, the FDA does not endorse a particular definition, but considers the scientific debate and context. Next slide, please. The definition of apathy has been evolving since the early 1990s, from a disorder of motivation to the most recent 2021 definition of clinically significant apathy specific to neurocognitive disorders. That definition addressed the lack of consistency between different prior definitions and scales. An International Society for Central Nervous System Clinical Trials and Methodology working group consisting of experts from academia, industry, and regulatory bodies developed a consensus definition using a modified Delphi model. That definition takes into account emerging knowledge of the neurobiology and circuitry of apathy and neurocognitive disorders, and the need to address observable traits in patients. It also considers the need to differentiate apathy from depression and other comorbid psychiatric, intellectual, physical, or motor illness, and from change in level of consciousness. The updated definition of apathy centers around symptoms and dimensions of diminished initiative, interest, and emotional expression or responsiveness. Next slide, please. In the year 2000, Jesty and Finkel proposed criteria for diagnosing psychosis in Alzheimer's disease, distinct from psychosis and schizophrenia in older individuals. Multiple subsequent diagnostic criteria have been proposed. Most recently, the International Psychogeriatrics Association initiated a consensus process that included input from clinical, regulatory, and industry stakeholders, and from a global network of experts in geriatric psychiatry, taking into account factors such as the diversity and temporal sequence of hallucinations and delusions in various types of neurocognitive disorders, as well as the variety of triggers, and the need to exclude many medical, neurologic, and psychiatric conditions. Criteria for psychosis across neurocognitive disorders were revised. The resulting criteria, which also extends to pre-dementia phases of neurocognitive disorders, define and give examples of hallucinations and delusions, which are the characteristic symptoms of psychosis. The criteria also clarify the duration and time course of symptoms and discuss exclusions, such as having previously met criteria for schizophrenia. Finally, associated features of agitation and/or depression were added to the revised criteria to account for the fact that psychosis in neurocognitive disorders is frequently accompanied by other neuropsychiatric symptoms. Next slide, please. In 2014, an International Psychogeriatrics Association working group engaged in a process that yielded a provisional consensus definition of agitation to enhance communication regarding this condition and to facilitate research. That definition views agitation as a distinct clinically identifiable syndrome, specifically in patients with cognitive impairment. The consensus process included a survey of possible behaviors to be included in the definition, such as pacing, verbal aggression, hitting oneself or others, and biting, among many others. The resulting criteria proposed that a patient must have at least one type of observable agitated behavior that can include excessive motor activity, for example, pacing, verbal aggression, for example, screaming, or physical aggression, for example, kicking or destroying property. Of note, the working group considered the relationship of agitation to aggression, which are overlapping but not identical concepts. The criterion that provides examples of aggressive behaviors allows for the presence or absence of aggression. The provisional consensus definition has been widely used in interventional and non-interventional research. Recently, the adjective provisional was removed from the criteria, but the individual criteria have remained unchanged. Next slide, please. The prevalence of neuropsychiatric symptoms and the temporal association with the severity of cognitive impairment differs based on the underlying etiology. Broadly, apathy, depression, and anxiety are among the most prevalent symptoms in Alzheimer's disease and other dementias, whereas elation and euphoria are the least prevalent. Apathy has been reported to be the most prevalent neuropsychiatric symptom in Alzheimer's disease and has been shown to increase with Alzheimer's disease severity. Agitation increases in prevalence with dementia severity and approaches 50% in community-dwelling individuals with severe dementia. Cumulative and persistent agitation has been reported to be as high as 60% in nursing home patients. The prevalence of psychosis varies depending on the underlying dementia type. It is reported to be highest in dementia with Lewy bodies as compared to Alzheimer's disease, for example. Delusions are more common than hallucination in Alzheimer's disease. Next slide, please. Neuropsychiatric symptoms are important to address because they are associated with poor outcomes, including faster functional decline and progression to severe dementia, increased hospitalization, morbidity and mortality, and earlier hospitalization in patients, as well as increased caregiver burden, distress, and lower quality of life. They are also associated with a higher cost of dementia care. Next slide, please. Non-pharmacologic approaches are recommended by various medical organizations and expert groups as first-line management for neuropsychiatric symptoms. Examples of non-pharmacological interventions include psychosocial approaches like music therapy and reminiscence therapy, sensory stimulation, and structured methods to assess and treat symptoms through modifying the environment. Medication use is reserved for distressing or dangerous symptoms that compromise safety. Various guidelines support off-label use of antipsychotics despite limited efficacy data and safety and tolerability concerns. Apart from antipsychotic medications, benzodiazepines, mood stabilizers, and antidepressants are used off-label. A significant challenge for developing recommendations for pharmacological treatment of neuropsychiatric symptoms is a small number of randomized clinical trials for the treatment of neuropsychiatric symptoms and dementia. Next slide, please. In 2023, the agency approved brexpiprazole for the treatment of agitation associated with dementia due to Alzheimer's disease. In 2016, the agency approved pimavanserin, the first drug to treat hallucinations and delusions associated with psychosis experienced by some individuals with Parkinson's disease. Of note, pimavanserin is not approved for the treatment of patients with psychosis unrelated to Parkinson's disease. The box warnings associated with these medications will be discussed in the upcoming presentation. there remains an unmet need for safe and effective therapies addressing agitation, psychosis, and other neuropsychiatric symptoms associated with cognitive decline of various etiologies. Next slide, please. In conclusion, neuropsychiatric symptoms are a heterogeneous group of non-cognitive symptoms which are very common in all types of dementia. They are associated with high rates of morbidity, mortality, and institutionalization, and carry a high burden for patients and caregivers alike. Management of neuropsychiatric symptoms is challenging, and current clinical practice guidelines are limited by lack of data from randomized controlled clinical trials. Various classes of drugs, including antipsychotic medications, are used off-label, and there's a high unmet need for safe and effective therapies.
Great. Thank you so much, Julia, for that wonderful presentation. We're going to do questions after the second presentation here. So we're going to go ahead and bring Shamir Kalaria, who's a senior clinical reviewer at the Division of Psychiatry within the Office of Neuroscience in the Center for Drug Evaluation and Research at the FDA, to sort of take us into the next steps. Shamir, go ahead.
Thank you, Nancy, and good afternoon, everyone. And thank you, Julia, for kicking us off and providing a great clinical overview of NPS. I think that leads us into this great discussion on the regulatory context and history of the antipsychotic box warning for the increased risk of mortality in elderly patients. So for the second half of this presentation, I would like to spend the next few minutes to present this historical overview, kind of the data that led to the box warning back in 2005, and to kind of shed some light in terms of the knowledge gaps that we've identified, some of the limitations of the analysis, and to discuss our future steps for upcoming analysis that the division and the agency plans to undertake. Next slide, please. So you would consider the signal actually to be almost a spur off of a previous analysis that was actually conducted by the agency starting in 2001. And back then, FDA had received a cluster of cases of serious cerebral vascular adverse events among subjects with dementia-related psychosis that were receiving antipsychotic treatment. And so while analyzing these cases, the agency did determine that there was an increased risk in cerebral vascular adverse events and issued warning statements for several atypical antipsychotics between 2001 and 2005. And these were for risperidone, olanzapine, and aripiprazole. Now, while looking into this data a little bit further and what the sponsors have provided with us in terms of their summary level data, what we also identified was that there was a potential increased risk of mortality amongst these patients that were receiving atypical antipsychotics. And therefore, we actually request additional sponsors to provide us data from their programs to further elucidate this signal. And so in 2005, the agency then conducted a meta-analysis to systematically look at this data and to quantitatively assess and estimate this increased risk of mortality. Next slide, please. So the agency's 2005 meta-analysis included 17 randomized short-term placebo-controlled trials that consisted of 5 atypical antipsychotics and 1 typical antipsychotic in elderly patients with dementia. As mentioned in the previous presentation, dementia was based on a clinical diagnosis in many trials that included patients with comorbid neuropsychiatric symptoms. The database included approximately 5,400 patients, 3,600 of which received active treatment and 1,800 of which received placebo. Now, the majority of this database, as you can see, included second-generation antipsychotics with the addition of haloperidol as the sole first-generation antipsychotic. And it was because that haloperidol was used as an active control in several of the clinical trials included in this database. It's also important to note that these trials were considered short-term studies, although they did range in duration between 4 to 26 weeks. The median duration of the 17 trials was typically 10 weeks in length. It's also important to note that there were no long-term studies included in this data, as most of the long-term studies came from open-label study designs that lacked a proper comparison arm. The average age of a patient included in these studies was approximately 80 years, with a range between 44 and 105 years in the study. In order to estimate the mortality risk, We obviously had to look at the frequency and the timing of deaths based off of clinical study reports and case narratives that were available at that time. But because the counts of death could vary based on the sampling timeframes, we actually evaluated differing sampling timeframes almost as a sensitivity analysis to assess the robustness of the signal. So what I mean by sampling timeframes, we looked at deaths that occurred within four days of treatment discontinuation, deaths that occurred within the intended period of observation, deaths that could have occurred within 30 days of the intended period of observation, and all deaths. Again, because patient-level data was not available at the time, it was also assumed that the follow-up amongst patients that either discontinued the trial for other reasons was also similar amongst those patients that had survived in the studies. Next slide, please. So the analysis ultimately revealed a 70% increased risk of death amongst subjects receiving antipsychotic treatment and placebo, and this is actually described in the current product label. Over the course of a typical 10-week trial, the incidence of death was 4.5% in the antipsychotic arm and 2.6% in the placebo arm. In the image on the right, it displays a forest plot describing the incident rate ratios for the various antipsychotics that were included in the meta-analysis. For this specific example, the incident rate ratio is based on the deaths that occurred within 30 days of the intended period of observation. Although the causes of death varied, most of the deaths appear to either be cardiovascular or infectious in nature, with many of the deaths being attributed to heart failure, sudden death, or even pneumonia. Now, because there were a limited number of well-defined cases based on the case narratives, while closely examining the case narratives, the specific mechanism by which the antipsychotic increased the risk of death still remains unclear today. Next slide, please. The graph on the left here describes the cumulative hazard of death over time based on subjects that were included in the 2005 database. As you can see, the hazard for death appears to be persistent over the course of the 100-day period that's described here and proportional between the two treatment arms. When you look at the figure a little bit more closely, the lack of concentrations of death that occur closer at the time of drug initiation within the first few weeks of treatment, this suggests that antipsychotics may not be directly the cause of death. And to kind of complement this finding, we actually went into the reports of the case narratives to look at the timing of deaths relative to preceding adverse events and the last dose of study medication, which also supported that the drug was usually not the direct cause of death, but may be also associated with worsening outcomes. The steady rise in the cumulative events with a higher rate in the antipsychotic group versus the placebo group also rather suggests that there was an indirect effect on death due to exogenous cases. Next slide, please. So based on these data, the agency required a box warning for all second-generation antipsychotics in 2005. I do want to highlight and emphasize that the agency did communicate these results of this meta-analysis in a drug safety communication that accompanied the regulatory action to issue the subsequent box warning. The results were also presented at the New Clinical Drug Evaluation, or NCD annual meeting, that also accompanied a consensus statement back in 2006. You can see the language for the box warning as it is applied to all the atypical and atypical antipsychotics on the right side of the slide. In 2007, the agency also reviewed 2 observational studies that suggested similar findings with typical antipsychotics, and thus then expanded the scope of the warning to include all antipsychotics by 2008. Due to the higher incidence of stroke and transient ischemic attacks, the agency also provided a class warning for cerebrovascular adverse events for all antipsychotics. It should be noted that at the time of the labeling change, no antipsychotics were approved for any of for treatment for any of dementia-related conditions. However, since 2005, there actually have been several programs that have evaluated the population that are similarly related that have led to further discussions on the application and the generalizability of the box warning. Next slide, please. So as Julia just discussed, pimavanserin was FDA approved in 2016 for the treatment of hallucinations and delusions associated with Parkinson's disease psychosis. Now, when evaluating the clinical studies that were submitted for marketing approval, the agency had similarly noted an imbalance in events between the treatment arms and determined that pimavanserin should also bear the box warning and precaution language about the use of this drug in demented elderly patients with psychosis. So you can see that the highlighted section in the box warning on the screen resembles closely to the existing box warning language for other atypical antipsychotics with several slight modifications. Next slide, please. In 2023, the agency had approved also brexpiprazole for the treatment of agitation associated with dementia of the Alzheimer's type. The results of the mortality analysis and the benefit risk discussion for this application was also discussed at a 2023 advisory committee meeting. Because brexpiprazole was approved for agitation without regards to the presence of psychosis, this resulted in concern about labeling, which could imply that in the absence of psychosis, the increase of mortality described in the box warning did not apply to the brexpiprazole treatment program for the treatment of agitation associated with Alzheimer's type. So during the review stage, ultimately, the review team ended up aligning the box warning with other antipsychotics and did not decide to recommend any additional changes. And you can see the modified language in the highlighted section for the box warning for brexpiprazole on the right. Next slide, please. Since the implementation of the box warning, the current literature includes various meta-analyses that further examine the risk of mortality amongst elderly patients treated with an antipsychotic. There were two specific meta-analyses that examined the same studies that were included in the agency's 2005 meta-analysis that further confirmed the described risk estimates in the product label. However, because the meta-analysis was mainly based on summary-level data, it remains unclear whether there are other demographic and risk factors, including the type and severity of NPS, that could contribute to the observed mortality risk. Therefore, to understand the potential risk factors and to further characterize this mortality risk, the agency has currently requested sponsors to submit subject-level data and plans to conduct a patient-level meta-analysis. And with that, I do hope that provides a glimpse of the historical background on the box warning, kind of what we've learned so far from the limited data that was available at the time, and a glimpse of our future plans. And I will turn it over back to you, Nancy, for any questions. Thank you.
Great. Thanks so much, Shamir. That was wonderful. We're going to turn to our question and answer, and we have about 10 minutes to address some clarifying questions that have come in. And just to acknowledge it, we do have other folks from the FDA. Julie and Shamir, if you want to come back on the screen for our question and answer, that would be great. We do have some other folks from the FDA that, you know, they are welcome to jump on and help with answering some of these questions. That would be fine too. But I think the first question I wanted to tap actually got answered in the question and answer a little bit, but I wanted to expand just a little bit on it. So the question was about using the term symptoms. And so I think this was more of a nomenclature question about symptoms traditionally referring to concerns expressed by the patient and signs to observable manifestations. And the suggestion from this participant that, you know, we might want to think about calling that signs and symptoms or manifestations, et cetera. And I did see that Valentina replied to that. So I didn't know if either of you had comments on that. But then in addition to that, I think this does highlight an even deeper question, at least in my mind, is that may, you know, share with us a little bit your thought on how this might impact how these different signs and symptoms are defined, because in this population, we do have unique challenges. We have patients with dementia who are often unable to tell us what they're feeling or what they're thinking or what they're going through. And so does that impact your thoughts in terms of the definitions here? So I think I'll start off with Julia, if that's okay, if you want to add anything to that. And then Shamir, welcome to jump in as well.
Absolutely. The question you raised is very important. And that's why the criteria include observable signs that don't rely as much on patient reporting, but absolutely that is extraordinarily important.
Shamir, did you have anything you wanted to add to that?
Nothing really to add. I think, again, I do recognize what the question has, and I think with these nuances, we're taking this into consideration, obviously, and I kind of agree with what Julie has to say. So nothing else to add.
Okay. And then we had another question from our audience, and I guess this one might be more to Julia as well. And the question was, although biomarkers allow for more definitive classification, did history, symptoms, and imaging provide some basis for classification?
So for the past clinical studies, clinical criteria were used for the classification. And notably, over the past decade, cerebrospinal fluid imaging and most recently plasma biomarkers have become available to allow us to determine whether individuals have Alzheimer's disease. And other biomarkers are available for other types of dementias.
Okay, great. This one I think goes to Shamir. This was with regards to the 2005 meta-analysis. And the question was, what were the exclusion criteria for patients in the study? So I don't know if they're referring to all of the studies that were involved in the meta-analysis, or if you did anything in terms of selecting patients from those studies. But either way, I think the question is valid in terms of what are the exclusion criteria, and specifically they're asking, were patients under hospice excluded?
This is a great question, and I think my response is going to allude to the heterogeneity that we're seeing across trials. Again, a majority of these trials were not submitted to us for review for a marketing application, and so this was kind of more in a post hoc in essence of requesting the data, what was already done, and so. In essence, the database and the meta-analysis was structured in the sense that we wanted to look at placebo-controlled randomized studies of similar duration. So that's kind of how we had included these studies in the first place. In terms of the definition of dementia, the criteria for specific neuropsychiatric adverse events, they may have varied across studies based on the instruments that were employed at that time. Some instruments used, such as the PANS, were used. There were CMAI used for other, symptoms of agitation to classify patients for inclusion and exclusion criteria. There were community dwelling patients. There were hospitalized patients included in the study. So this we could consider it almost a kind of an all-comers analysis. And then the difficult aspect of this, kind of to understand, where there are specific patients at risk for, mortality. It's difficult to say because this was all based on summary level data. We didn't really have patient level data at the time. And so that kind of goes into kind of our next steps. If we were to obtain this patient level data, what would be some of those variables relating to the eligibility criteria and then the patient population that we could possibly use to understand the risk further. So I hope that answers the question.
Yeah, terrific. Yeah, and I think we'll touch on this a little bit in session 4 today as well when we start thinking about some of those next steps. So that's terrific. Julie, I think this one is probably more geared to you. It's given that agitation is common across dementia types, how generalizable are clinical trial safety results across neurocognitive disorders?
Thank you. The generalizability of safety data is unclear, and that's partly because the criteria for defining Alzheimer's disease and other neurodegenerative disorders has evolved. And the diagnostic criteria for certain neuropsychiatric symptoms like psychosis have been developed.
Okay, great. And I guess now that we're sort of talking about generalizability, Shamir, this is, I think, for you, is what is the generalizability of the boxed warning to patients with different comorbid neuropsychiatric symptoms or forms of dementia?
Again, a great question. And I think this also goes back to the 2 cases that we were discussing, pimavanserin and brexpiprazole. Specifically with brexpiprazole, that was looking at agitation, but we do understand that there's a high degree of comorbidity between all these neuropsychiatric adverse events. So it is something that we're considering. Again, the difficult aspect of this is kind of how do we understand the population makeup of those that were included in the 2005 database. A lot of these patients did have comorbid neuropsychiatric symptoms. So it's hard to say whether or not patients that had higher severity of psychosis were those that were at risk, when we really can't quantify that with the data that was available at the hand. So we do recognize that these symptoms do occur comorbid amongst this patient population, and that is something that we're looking to investigate further.
Okay, terrific. And I think, Shamir, also, you talked about the risk of causes of death in the various studies, and I think that's a really important question. I mean, I remember too, as a practitioner, when we first heard about this, since I was in cardiology, we were thinking QT prolonging, and all this sort of stuff. But can you elaborate any more on mortality risk difference based on the cause of death at this point?
Again, as I mentioned before, the difficult aspect of understanding the cause of death is dependent on how well-defined the cases that were submitted to us. And so based on the limited information at the time and what was included on the case narratives, it's very hard to attribute a single adverse event or a fatal adverse event in these cases. Now, the review team at the time tried to characterize or categorize the causes of death based on cardiovascular nature, infectious nature, or even other causes. And what was identified was between the different, amongst patients that had cardiovascular death or those that had death due to infectious nature, the incident rate ratio didn't differ across different treatment groups or different the different antipsychotics, which is a good thing because it's consistent. But it's tough to say whether or not the risk was elevated specifically for patients that had cardiovascular disease or those that had background comorbidities that could have attributed to this higher risk for this adverse event. So that is, again, limited to the data that we had at the time. And it's also, again, it's one of the strongest limitations of this is just strictly the amount of information that was included in the case narratives at that time, too.
Okay, terrific. Yeah, and there's a lot of great questions in our Q&A, and I think that a lot of them highlight some of the heterogeneity that we're talking about here, sort of the subsets of populations. We've got questions about home versus in care facilities and those sorts of things. So I think those are all great. We can encourage the audience to keep sending those because those will all be helpful in the long run as we're trying to think through what the audience thoughts are and what might be appropriate to consider here. I think in the last minute we have, there was a clarifying question that came in from someone, and Shamir, I don't know if this is something you can answer or not, and quickly, it's, can you let us know where you obtain the safety data for Nuplazid to determine the imbalances in mortality?
So for the specific indication, we did receive the clinical studies that were intended to support evidence of effectiveness and safety. And so the imbalances were observed in those studies, in addition to other external data that was also used for support that development program.
Okay, terrific. All right. Well, that was great, and really appreciate everyone submitting their questions. Very, very helpful in getting your thoughts on where some of these questions or issues need to move as we move forward. So thank you both, Julia and Shamir, for participating in the session and for this great start of our workshop. I'm now going to turn things over to my colleague, Christina Silcox, who will be moderating our next session. Christina, take it away.
Thank you, Nancy. And also thanks to Shamir and Julia. What a great opening session. My name is Christina Silcox. I'm the Research Director for Digital Health at the Duke-Margolis Institute. And we are now going to move on to our next session of the day, titled Review of Data Analysis and Literature Review Findings. We will follow the format of the first session, where we will have two presentations followed by a brief Q&A with clarifying questions from the audience. As a reminder to the audience, you can go ahead and submit questions through the Zoom Q&A function throughout this session. And with that, I'd like to welcome our first speaker, Funyang Zhao, mathematical statistician in the Division of Biometrics 7, Office of Biostatistics at the FDA. Funyang, I'm going to turn it over to you now.
Thank you, Christina. Hello, everyone. I will be presenting our recent meta-analysis on the use of antipsychotic and associated mortality risk in elderly patients with dementia-related psychosis and other behavior symptoms. Next slide, please. This new meta-analysis was conducted using a systematic approach on the data submitted to the agency in 2005. Our primary goal was to assess the mortality risk associated with antipsychotic drugs in randomized placebo-controlled clinical trials. These trials were designed and conducted to evaluate the safety and effectiveness of antipsychotic on psychosis and other behavior symptoms in elderly patients with dementia. Next slide, please. This slide outlines the trial site included in our meta-analysis. In total, we analyzed the same 17 randomized placebo-controlled trials as the 2005 analysis that examined the use of antipsychotic medications in elderly patients with dementia-related psychosis and other behavior symptoms. As shown in the table, these 17 trials investigated 5 atypical antipsychotics: aripiprazole, olanzapine, phenterpine, risperidone, and ziprasidone, alongside haloperidol, which is a typical antipsychotic and has served as an active control in some trials. The treatment duration across these studies range from 4 to 26 weeks, with the majority of trials having a duration of between 8 and 12 weeks. The sample size of the study population includes a total of 5,377 subjects. Of this, 3,611 participants were assigned to the antipsychotic drug groups, while 1,766 participants received a placebo. In terms of data available for analysis, 7 out of the 17 trials provided patient-level data for olanzapine and risperidone. The remaining 10 trials included summary-level data. Additionally, as indicated in the dose of treatment column, some trials include multiple dosing treatment groups, and a few trials incorporated an active control group. Next slide, please. This slide provides the statistical approach used in our meta-analysis. Our primary endpoint was all-cause mortality. This means the analysis includes the deaths from any cause among the participants. We compared antipsychotic drug groups with the placebo groups within each trial, multiple doses of the investigational drug and an active control drug, if used, such as haloperidol, were put together as a drug group. Regarding our analysis population, we included elderly patients with dementia-related psychosis or other behavior symptoms who were enrolled in the 17 randomized placebo-controlled trials. We use an on-treatment analysis approach to handle treatment discontinuation. This method focuses on the period during which participants were actively receiving the treatment. We included deaths that occurred within 4 days of treatment discontinuation. And we acknowledge limitations of the use of an on-treatment analysis, and this will be discussed later in the presentation. However, the choice of an on-treatment analysis was based on the data available, for which we were confident that mortality information was consistently captured across all 17 trials. It is less clear if participants in the trials were followed treatment discontinuation to support an on-study analysis that would include all deaths until trial end. Next slide, please. This slide describes how we ascertained the mortality in our meta-analysis. Our approach was grounded in a clinical trial of case narratives for all deaths that occurred within the included trials. In this case narrative review, we focus on capturing 3 key time points: first dose date, last dose date, and death date. All deaths that occurred within 4 days of the last treatment date were counted in the on-treatment analysis. And this approach is consistent across all 17 trials. Next slide, please. This slide explains how we calculated the exposure time to antipsychotic medications in our meta-analysis. To ensure accuracy, we differentiated between 2 types of data available from the trials. Firstly, for the 7 trials that provided patient-level data, we had detailed information on each participant. In these cases, drug exposure was calculated starting from the dose start date. We determined the follow-up period for each patient by censoring it at the earliest of 3 events: the dose end date plus 4 days, the date of death, or the end of the double-blind study period. For the 10 trials with only summary level data available, we extracted the total number of drug exposure days for each treatment arm directly from the study reports. By using this approach, we were able to integrate both patient-level data and the summary-level data. Next slide, please. This slide explains the statistical methods we employed in our meta-analysis to assess the mortality risk associated with antipsychotic use. Our primary statistical metric is the incidence rate, calculated as the number of deaths per 100 patient years of drug exposure. We estimated the incidence rate difference between the drug and the placebo groups using a random effects meta-analysis model. Additionally, we calculated the incident rate ratio along with its 95% confidence intervals using a negative binomial model. This model is used to handle zero-event trials where no deaths were captured in either the retreatment or placebo groups. Furthermore, we also assess study heterogeneity to determine the consistency of results across different studies. We measured heterogeneity using the I-square statistic, where a smaller I-square value indicates less heterogeneity. Next slide, please. This table shows the aggregate summary of patient demographic across all 17 trials. As shown in this table, the average age of participants is approximately 80 years. Regarding gender distribution, around 70% of participants are female, with similar percentages observed between the treatment and the placebo groups. The racial distribution is also comparable across both treatment and the placebo arms. And the baseline MMSE scores average around 11. And these scores are comparable between the treatment and the placebo groups. In summary, the demographic characteristics across the trials are well balanced between the treatment and the placebo groups. Next slide, please. This table presents the average exposure time for each trial, calculated by dividing the total exposure time by the number of patients. Our purpose here is to assess whether there are differences in average treatment time between the treatment and the placebo groups within each trial. Due to the lack of patient-level data across all studies, we rely on summary data to provide average treatment exposure time for each group. As shown in this table, we didn't find any noticeable or consistent difference in the average treatment exposure time between the treatment and the placebo groups. Next slide, please. This table presents the key results of our analysis. The event column shows the number of deaths in both the treatment and the placebo groups across all included trials. And PY represents the total patient years across all trials. Here, we calculated the incident rate for both the treatment and the placebo group. To compare these rates, we estimated the incident rate difference using a random effect meta-analysis with inverse variance weighting, resulting in an incident rate difference of 4.7, so per 100 person year. So roughly speaking, this means that if 1,000 patients were treated for 1 year, we would expect to see 47 additional deaths if treating with an antipsychotic rather than placebo. The 95% confidence interval is 1.6 to 7.9, meaning the difference in deaths could be as high as 7.9 per 100 patient years. Furthermore, we estimated the incident ratio using a negative binomial random effects model, which yield an incident rate ratio of 1.76, suggesting a 76% increased risk of death among subjects receiving antipsychotic treatment versus placebo. And the lower bound of confidence interval is 1.02. So in summary, both estimates of the incident rate difference and the incident rate ratio excluded a value of no effect, suggesting an increased risk of death in the treatment group compared to the placebo group. Next slide, please. This figure presents the forest plot for the incident rate difference based on a random effects meta-analysis using inverse variance weighting. Based on the incident rate difference forest plot, risperidone study USA 63 has the highest total number of death events. And Quantabine study 5077 US has the largest incident rate difference effect. And there are 4 trials, 1 trial of olanzapine, 1 trial of Quantabine, 1 trial of risperidone, and ziprasidone demonstrate an incident rate difference effect of less than 0. Additionally, the I-squared statistic is low, implying minimal heterogeneity among the studies. So this low level of heterogeneity suggests that the included studies are relatively comparable. Next slide, please. And this figure presents the forest plot for the incident rate ratio based on a negative binomial random effects model, which effectively accounts for 0 event trails. So based on the incident rate ratio forest plot, risperidone study USA 232 has the largest incidence rate ratio effect. And 4 same trials demonstrate an IRR effect of less than 1. Overall, the direction of the effect size is consistent with the incident rate difference forest plot presented on the previous slide, except that the trials with 0 events in either arm are not summarized in this incidence rate ratio forest plot. Next slide, please. To summarize our meta-analysis across 17 trials, there were 76 deaths that occurred within 4 days of treatment exposure. Among all mortality events, there are 59 deaths out of 3,611 participants in the antipsychotic group, and there are 17 deaths out of 1,766 participants in the placebo group. The meta-analysis suggests an increased risk of death when treating with an antipsychotic compared to placebo in elderly patients with dementia-related psychosis and other behavior symptoms. The estimated incident rate difference is 4.7 per 100 patient years, with a 95% confidence interval of 1.6 to 7.9. Additionally, the estimated incident rate ratio is 1.76, with a 95% confidence interval of 1.02 to 3.04. Next slide, please. So here, we would like to highlight several key points in our meta-analysis. Firstly, our analysis employed an on-treatment approach due to the lack of data following treatment discontinuation. The existing data does not support a reliable method for conducting an on-study analysis consistently across all 17 trials. Given this, it is important to acknowledge several limitations. The on-treatment analysis may break the integrity of randomization and introducing potential bias. Differences in treatment discontinuation time and the characteristic of participants who discontinue treatment between arms can affect the comparability of these groups. As such, this may compromise the validity of our mortality risk estimates. The restricted availability of data limits our ability to perform a more comprehensive assessment. Additionally, the absence of subgroup data prevents us from identifying potential populations that may be at increased risk when using antipsychotics. In summary, while our findings indicate a significant increase in mortality risk associated with antipsychotic use, these limitations highlight the need for cautious interpretation of our study findings and the importance of future research to address these gaps. Next slide, please. Finally, we want to discuss the future steps based on our current findings. The FDA has issued an information request to individual sponsors seeking patient-level data from each of the 17 trials included in our study. Obtaining this detailed data is essential for conducting a more detailed patient-level analysis. With access to patient-level data, our primary intent is to assess the mortality effect through the end of the double-blind treatment period, effectively performing an on-study analysis. Furthermore, the availability of patient-level data will enable us to explore specific subgroups of patients who may be at increased risk. These further steps are important for enhancing the robustness of our findings and addressing current data limitations. That is the end of my presentation. Thank you for the time.
Thank you so much for such an informative presentation. We're going to now turn to our next presentation. I'd like to welcome Jessica Young, a clinical reviewer with the Division of Neurology 1, Office of Neuroscience within the Center for Drug Evaluation and Research at the FDA. Jessica, go ahead and get started whenever you're ready.
Thank you, Christina. Good afternoon. In this presentation, we'll focus on observational studies evaluating the mortality risk following antipsychotic exposure in patients with dementia. Next slide, please. The objectives of this talk are to review and summarize findings of the available observational studies to complement knowledge generated by the meta-analysis of prior randomized controlled trials, with a focus on mortality risk and potential factors contributing to mortality risk in patients with dementia following antipsychotic exposure. Next slide, please. Randomized controlled trials are the ideal type of study to address questions related to mortality risk in patients with dementia who are exposed to antipsychotics, given reduced bias with randomization. However, the available data set from prior randomized controlled trials has a few limitations. These randomized controlled trials were generally small studies, mostly including 200 to 300 participants. The findings may not be generalizable, as the studies used highly selected samples based on study-specific inclusion and exclusion criteria. And many of the studies were done in patients residing in nursing homes with varying degree of dementia severity. These studies had a short duration of observation, typically lasting less than 12 weeks. The rate of death was generally low, requiring combined data and meta-analysis to estimate the mortality risk. Next slide, please. To complement our understanding of mortality risk from the prior randomized controlled trials, we reviewed observational studies, many of which generally include population-based samples with larger sample size and longer observation period. We searched for observational studies, including search term death, mortality, antipsychotics, dementia using PubMed Medline. We limited our search to studies published after 2008, when the second box warning was issued. Of the 37 articles from the search, 15 articles that focused on the relationship between overall all-cause mortality risk and exposure to conventional and/or atypical antipsychotic as a class in patients with dementia were selected as the main articles for further review. Studies examining mortality risk of individual antipsychotics or comparative risk of an antipsychotic related to a reference drug were excluded. Next slide, please. First, we wanted to see if observational studies show similarly increased mortality risk in patients with dementia taking antipsychotics as the prior randomized controlled trials. Next slide, please. 9 studies published between 2011 and 2022 showed higher overall mortality risk in patients with dementia and antipsychotics, whereas 5 studies published between 2014 and 2022 reported no statistical difference in mortality risk in these patients. 1 study from 2018 reported lower mortality risk. These studies generally had similar characteristics. They were mostly cohort studies using national registry, claims data, prescription data, health records, and research database as data source. Studies included conventional and/or atypical antipsychotics. The follow-up duration varied anywhere between 180 days and 10 years. Many of the studies demonstrating higher mortality risk used population-based cohort with larger sample sizes, whereas studies suggesting no increased mortality risk relied more on regional samples with relatively smaller sample size. Next slide, please. This table summarizes the 9 studies that showed higher mortality risk following antipsychotic use in dementia, as well as the 6 studies that demonstrated no statistical difference in mortality risk or lower mortality risk. I'll summarize the findings in the next slide, but the table is here for your future reference. Next slide, please. Of the studies evaluating overall all-cause mortality risk following antipsychotic use in patients with dementia, most studies found the risk to be higher, with a hazard ratio ranging from 1.18 to 3.7 when using Cox regression models. Of the studies that demonstrated no statistical difference in mortality risk in patients with dementia exposed to antipsychotics, 3 studies estimated hazard ratio to be 0.94 to 1.06, all of which had 95% confidence intervals, including 1. In one study, the difference in mortality risk became no longer significant when an adjustment was made for cognitive status along with other patient characteristics. In another study, a similar observation was made after adjusting for neuropsychiatric symptoms. One study found that the mortality risk was lower in Taiwanese patients with Alzheimer's dementia who were exposed to antipsychotics than those who were unexposed, with the hazard ratio of 0.66. Next slide, please. However, cross-study comparisons of these observational studies are challenging due to variations in study design and analysis. Most of these studies were conducted in a single country, and therefore the findings may reflect unique aspects of clinical care in a given country. Clinical and demographic characteristics of the samples are different across the studies. Sample sizes, including the exposed and unexposed groups, also vary. In addition, the duration of exposure to antipsychotics and follow-up differ across the studies. When studies used the Cox proportional hazards model, covariates that were used for adjustment were different across the studies. Next slide, please. Despite the heterogeneity of the observational studies and some inconsistent findings, overall, the mortality risk appears to be increased in patients with dementia who are exposed to antipsychotics, similarly to the findings of the prior randomized controlled trials. But does the mortality risk change over time in these patients? It's difficult to ascertain this information using the randomized controlled trials, as these studies typically had a short duration of follow-up. Several observational studies evaluated long-term mortality risk following antipsychotic use in dementia. Next slide, please. Most of these studies examined short-term versus long-term risk over 180 days, but some studied it up to 6 years. The mortality risk in patients with dementia on antipsychotics appears to be higher short-term, especially in the first 30 days. In one study indicated no increased mortality risk after 30 days, whereas other studies show a decrease in mortality risk after 30 days compared to the first 30 days, although the risk may persist long-term. Greater risk of mortality early in the treatment course may suggest that there may be unmeasured medical confounders that are associated with the need for antipsychotic use in patients with dementia. Lower risk with longer durations of treatment may also reflect survivor bias. Next slide, please. The reasons why patients with dementia are prescribed antipsychotics. Question is, whether these factors that require the use of antipsychotics in patients with dementia play a role in mortality, or antipsychotic are an independent contributing factor. Next slide, please. One of the potential risk factors for mortality may be the severity of dementia. Limited studies have examined the effects of dementia severity on mortality risk following antipsychotic use. When mortality risk was assessed in patients with dementia exposed to antipsychotics in observational studies, not all studies adjusted for dementia severity to estimate the hazard ratio. When the studies adjusted for dementia severity, they used indirect measures such as age at dementia diagnosis and duration of dementia. In one study where the mortality risk was estimated after adjusting for the severity of dementia using measures of cognition and function, such as the mini-mental state examination and activities of daily living scores in community dwelling patients with mild to moderate Alzheimer's dementia, the use of antipsychotics was no longer associated with increased mortality risk, although the model was also adjusted for other patient characteristics. Therefore, the changes in risk estimate in this study cannot be solely attributed to dementia severity, although dementia severity may still play a role. Next slide, please. Another factor to consider is the type of dementia. One study showed increased all-cause mortality in patients with Alzheimer's dementia or vascular dementia who were exposed to antipsychotics compared to those who were not exposed, but not in patients with mixed-type dementia or unspecified other dementia. When testing for interactions between antipsychotic use and type of dementia, the increase in mortality risk strengthened in vascular dementia, but not in Alzheimer's dementia. These findings suggest that each dementia may differentially affect the mortality risk associated with antipsychotic use. But overall, there are insufficient data to draw meaningful conclusions, as most observational studies do not specify dementia type included in their studies. And even when specified, it's difficult to determine diagnostic accuracy given the data sources that are typically used for these studies, such as claims data. Next slide, please. Similarly, limited studies have investigated the contributing effects of neuropsychiatric symptoms on mortality in patients with dementia who are exposed to antipsychotics. In one study, antipsychotic-related mortality was not increased when adjusted for neuropsychiatric symptoms for which treatment was prescribed in patients with probable Alzheimer's dementia, suggesting neuropsychiatric symptoms may be a significant predictor of mortality. In general, neuropsychiatric symptoms may influence mortality in patients with dementia, as studies shown an increased risk of mortality associated with moderate or severe behavioral and psychological symptoms of dementia, as well as earlier death in patients with Alzheimer's dementia who experienced psychosis, agitation, aggression, and affective symptoms. However, no clear interactions between neuropsychiatric symptoms and antipsychotic use in relation to mortality have been demonstrated in patients with dementia. Next slide, please. In a study using a sample of patients with Alzheimer's, vascular, mixed, and other unspecified dementia from UK, an increase in overall mortality and stroke-specific mortality was noted in the whole sample when adjusted for socio-demographic characteristics, dementia type, cognitive and neuropsychiatric symptom scores, and prior hospitalization. However, when mortality was examined based on the presence or absence of agitation and psychosis, no increase in overall or stroke-specific mortality risk was observed in patients with dementia who were exposed to antipsychotics compared to those who were not exposed. In addition, no significant interaction between neuropsychiatric symptom strata and antipsychotic use in relation to mortality was noted. Based on the available data, at this time, It's difficult to determine whether antipsychotic use is an independent predictor of mortality, or whether other factors such as the underlying dementia or neuropsychiatric symptoms play significant roles in determining mortality risk in patients with dementia who are exposed to antipsychotics. It may be possible that multiple factors converge to contribute to an increased mortality risk in these patients in a complex manner. Next slide, please. As previously stated, there are several limitations to literature review using observational studies. The most important limitation is the potential for biases due to the lack of randomization in observational studies. Although not specific to observational studies, publication bias may influence the review of the available data. Potential unmeasured confounders can also be introduced. Variations in data source, sample, study design, and analytical method make cross-comparisons of these studies difficult. Next slide, please. Based on the available literature, the mortality risk following antipsychotic use appears higher in patients with dementia. The risk may be greater short-term, although survivor bias may be present. There may be other factors that may refine the relationship between antipsychotic exposure and mortality risk in patients with dementia. Nevertheless, it remains challenging to draw meaningful conclusions in the presence of conflicting findings and limitations of the observational studies. Internal validity of the observational studies will need further assessment. Thank you.
Thank you, Jessica, for walking us through that information. It was really a wonderful presentation. So once again, we're going to turn to the audience to ask clarifying questions about the presentations. I have been given a note that other FDA speakers online may pop in if appropriate. And if they do, you're welcome. Please just make sure that you introduce yourselves when you come on video. So we already have a couple of really great questions from the audience members. To start with, Feng-Yu, given that neuropsychiatric signs and symptoms of dementia are an end-of-lifespan phenomenon, and dementias are commonly terminal, it could be helpful to also understand the impact on life expectancy if the risk of death is not concentrated in initial treatment, but appears to be more uniformly distributed. Has such an estimation been undertaken?
Thank you for the question. So this analysis is our first step to reevaluate to see if there is a signal. So we have not undertaken such analysis.
And then I have a question for Jessica. In the observational study review, how is confounding by disease severity addressed when comparing atypical antipsychotic use to non-use of any treatment?
That's a good question. As I briefly mentioned, there are a lot of heterogeneity in these observational studies. So when it comes to dementia severity, some studies use age at onset of dementia or as sort of a surrogate marker. Some studies did use cognitive measures as an adjusting factor. When it comes to neuropsychiatric symptom severity, sometimes some studies use a scoring method to determine mild versus moderate or severe, but some studies, it's hard to sort of elucidate how they adjusted for those confounding factors.
And for Feng-Yu, we have one question from the audience. Didn't the 2005 analysis include an analysis that suggested that deaths were reliably reported up to 30 days beyond the intended period off treatment, allowing an on-study analysis that showed similar results?
Thank you for the question. That is a great point. So the 2005 analysis did include an assessment, provide summary reports up to 30 days, like beyond the treatment discontinuation. However, so actually the data after treatment discontinuation are still uncertain. For example, people may lose contact after treatment discontinuation, and this is not considered in March data summary report. And it's important to note that this data set are from 20 years ago, and the clinical guidance have evolved a lot since then. So to ensure that our it fits to our current on-study analysis, we aim to obtain the patient level data to support our future on-study analysis.
That makes sense. And then Jessica, did the mortality risk relate to dose? Was the dosage level available in any of these studies?
So dosage level per se weren't available in any of the studies that I reviewed, but they did, some of the studies sort of calculated daily equivalent dose level, factoring duration of exposure. But again, those studies are quite limited, and so we don't have a clear understanding of what the dose is doing in terms of mortality risk.
Okay. And then this next question is for both of you. Could you speak to any industry collaboration that may be supportive of improving data available for research? Kind of a stepping back type question. Can we start with you, Feng Yu?
I think I'll defer this to the panelists because that's like a future collaboration.
Okay. And then I think a kind of a related question. One participant commented that patient-level data is the desirable data collection type, and asked what key characteristics should the optimal data collection system include that are unique to neuropsychiatric outcome studies?
Yeah, I think patient-level data is very important to support the meta-analysis, and I can answer from one perspective. So I think patient-level data, they should capture the detailed information on demographic of patients, also the medication use and some comorbidity conditions, and it should also have a comprehensive baseline assessment and follow-ups to track changes over time. So for example, we want to know the date of treatment discontinuation and the date of last contact of the participants, and that will support our future analysis.
Okay. And Jessica, can you speak more to what might be the most, and this kind of relates to what I just, the answer that Feng Yu just gave. Could you speak a little bit more to what the most important potential confounder in the observational studies reviewed that limits the interpretation of the studies?
Yes, so the underlying dementia, including severity, may be one of the most important confounders. As previously discussed, we have limited data on what role dementia plays in mortality risk following antipsychotic exposure. Many of the available studies either didn't adjust for dementia severity or didn't use cognitive measures to adjust for severity. Looking at dementia severity in real world setting may be challenging, as clinical settings, evaluation tools, and criteria may vary, so it may be difficult to control for it. But it will be important to understand how it contributes to the risk of mortality. And we'll have further discussion on future opportunities in a later session.
Oh, good. Great. So I just want to remind the audience that if you have questions, clarifying questions for the two speakers, please do go ahead and put them in the Q&A box. I do have another one for Feng Yu. If you could speak more to any subgroup analysis that was conducted to assess populations at higher risks.
Thank you for the question. So currently, we did not perform a subgroup analysis to assess populations at higher risk. This decision was due to due to the data limitations, because for now, as only 7 out of 17 trials provided patient level data. However, there are still future opportunities to explore the question when we have more comprehensive patient level data become available. And this analysis will incorporate clinical input and careful considerations to identify potential higher risk populations.
And speaking of the meta-analysis, that did that include studies conducted between 2005 and now? And if not, why not?
So it doesn't include studies between 2005 and now. It only includes studies submitted in 2005. Part of the reason is that we want to make sure the demographic and the characteristic of these studies are comparable. If you for example 20 years ago trial and the current trials, they may have different characteristics. So if we want to do meta-analysis, we want to make sure they are comparable, and we want to double check the data set from the current studies before we conducting integrate the meta-analysis together.
And I think we have maybe one more question. Hold on one second. So this is so this again is for both the speakers. You both covered a lot of content during the session. I really appreciate that. Are there any additional comments that you'd want to make before we move to the break? And Jessica, maybe we can start with you.
Sorry, I was having a hard time unmuting. Observational studies help us understand some of the questions that may be hard to address using randomized controlled trials, but there are a lot of limitations and there's still unanswered questions. So there are going to be further work and future opportunities to answer some of the questions, especially considering are there modifiable factors or contributing factors to this mortality risk in patients exposed to antipsychotics?
Thank you. And actually, before we go into Feng Yu's last comment, we actually did get a couple more questions come in. One is, are there any specific neuropsychiatric symptom domains that showed as a signal, as a predictor of risk in any of the meta-analysis or the observed studies? So I think this is a question for both of you.
At least from the observational studies, again, the studies looking at specific neuropsychiatric symptoms and its interaction with antipsychotic use for mortality have been limited. And the available studies so far have not shown any interactions between specific neuropsychiatric symptoms and antipsychotic use for mortality, as discussed before during the presentation. But one thing to keep in mind is that, again, the data are limited and there are some limitations to the observational studies.
Mm-hmm. And did you have anything you wanted to add to that?
So currently we don't have enough data to assess this in our meta-analysis.
And then one attendee commented that confounding by indication is a major problem in a non-randomized context. Agitation and insomnia may be expected to correspond to increased psychological stress and immunosuppression, potentially increasing the risk for death. Could treatment response help ameliorate this problem?
I think the review that we did really just focused on specific questions about mortality risk in antipsychotic use. The studies have not really gone into more detail of other characteristics like insomnia or immunosuppression. So it's difficult to answer that question based on the studies that's been reviewed.
All right, so I do want to go back now and give Feng Yu a chance to make any additional comments. And I'm actually also getting a note that Valentina may want to jump on with some additional clarifying points after your comment.
Sure. Thank you. So I just want to say this is our this analysis is the first step to confirming the mortality risk associated with antipsychotic use, and we confirm the signal consistent with previous findings based on our current data availability and we want to still do some like further analysis when more patient level data available. So yeah, that's the comment from me. Yeah.
Thank you so much. And Valentina, did you want to add anything?
Well, hello, this is Valentina Mantua. I am the clinical team leader, Division of Psychiatry, and I would like to thank the audience for the very many questions. We weren't able to answer all of them, but most of the questions were related to confounding factors, both for the randomized controlled trials as well as the observational studies that we've summarized, but we haven't really done a meta-analysis as for now. And I would like to say that we will address, later in the panel, these outstanding questions, which we do have ourselves. So you've heard about the limitations of the data, but one thing we definitely want to look into is what is the contribution of the type of diagnosis, of the type and cluster of neuropsychiatric symptom and the severity, and potentially also the length of treatment. So this is something we're going to be looking at, and what is clinically relevant are outcomes related to these variables. So if more data will come in, we hope that we will have a patient level definition of this variable in order to be able to do everything that you've suggested correctly. I just wanted to point that out.
Thank you. Thank you so much. So I think that's all the time we have for this session, but I do want to just thank Jessica and Feng Yu again for such great both presentations and additional detail through questions, and Valentina for jumping on at the end and giving some additional clarifying remarks as well. Hello, everyone, and welcome back to our meeting on mortality and antipsychotic use in dementia-related behavioral disorders. In this third session of the afternoon, we're going to be covering stakeholder perspectives on considerations regarding the use of box warnings for antipsychotics. Panelists will share perspectives and considerations related to the increased risk in mortality and the use of antipsychotics in dementia-related behavioral disorders, particularly in older adults. Before we turn to the panel discussion, we're going to be joined by Daniel Weintraub, Professor of Psychiatry at the University of Pennsylvania, who will be providing a brief presentation to help us get started. Daniel, if you could please join us on the screen at this time, I'm going to turn things over to you.
Thank you, Christina. Hello, everyone. I was given the daunting task in 10 minutes of presenting some clinical research perspectives on the use of antipsychotics in neurodegenerative diseases. This is brief then. It's not going to focus much on mortality because you've already heard a lot about that. And I'm going to shift a little bit the focus from just Alzheimer's disease to neurodegenerative diseases more broadly. Next slide, please. So a little bit about Alzheimer's disease first. Next. So it's clear that if you look at clinical trials, randomized controlled trials in different ways, that antipsychotics appear to at least be efficacious for the treatment of psychosis and or agitation. This is a randomized discontinuation study that was done some time ago, but really makes the point that antipsychotics seem to be efficacious. Next slide. I like referring to this study led by Dr. Lon Schneider called the CATIE-AD study. And the reason I like it is because it really informs our clinical management, where it looks at not just the efficacy of antipsychotics, but their effectiveness. So balancing the side effects, adverse events versus the beneficial antipsychotic effects of antipsychotics. And here, what we learned from this study was that adverse events offset the advantages and the efficacy of atypical antipsychotic drugs for the treatment of psychosis or agitation in Alzheimer's disease. A little more detail to follow, so next slide. So the time to discontinuation or treatment discontinuation in the study for lack of efficacy favored the antipsychotics that were used in the study, particularly risperidone and olanzapine, to atypical antipsychotics. And that was reflected in the hazard ratio as well. So this is efficacy. Next slide. But then if you look at the treatment discontinuation by intolerability or adverse events, that's the other side of the effectiveness story, that the time to treatment discontinuation favored placebo in this case. So all three active compounds, olanzapine, risperidone, and quetiapine, had higher rates of treatment discontinuation due to those reasons. If you get into the details, which is what you have to deal with on a clinical basis day by day. There were higher rates of Parkinsonism and extrapyramidal symptoms in 2 of the active treatment groups. Sedation was more common in all 3 antipsychotic groups. And then there was also confusion and mental status changes, more common with olanzapine and risperidone. In addition, for atypical antipsychotics, from a clinical standpoint, you have to worry about things such as increased risk for type 2 diabetes, orthostatic hypotension, dry mouth, dizziness, and constipation. Next slide. There's also concern, perhaps, and this is also data from the KDAD study looking at a cognitive summary score, that antipsychotics may adversely impact cognition in older patients, dementia patients. Next. And then just a brief mention of mortality. Obviously, that's a focus for this whole day or half day, but there is clear risk, as you've heard about, with increased mortality in Alzheimer's disease. This was the DART-AD study, another randomized discontinuation study, showing that those patients who remained on an antipsychotic had higher mortality risk than those that discontinued antipsychotic treatment. Next. So I do want to shift a little bit to other populations, because I think actually the population I work most closely with now is Parkinson's disease and to some extent dementia with Lewy bodies. Their psychosis is a little bit of a clearer picture, and it's also a more complex illness. So I think it's actually more informative in some ways than just looking at Alzheimer's disease. Next. So we know that within Parkinson's disease, and this would be both Parkinson's disease with and without dementia, so this really broadens the conversation some to more about neurodegenerative diseases broadly, not just dementia, that PD psychosis is a problem in terms of long-term care, hospitalization, so some of the same things you heard about for Alzheimer's disease already. Next. We also know that it appears to be an independent risk factor for mortality. So psychosis in and of itself is a mortality risk. Next. We had done some analyses previously using Department of Veterans Affairs data looking at antipsychotic use in Parkinson's disease, and similar to what's been reported for Alzheimer's disease. We saw an increased risk of mortality at 6 months. These are observational data for both atypical antipsychotic use and typical antipsychotic use as well. Again, similar to what's been reported for Alzheimer's disease. And again, these were not all Parkinson's patients with dementia. It was just Parkinson's disease patients more broadly. Next. You heard a little bit about pimavanserin already, and since it's in Parkinson's disease and that's my area of focus, I just want to highlight a couple of things about it. So one difference is that this is a pure serotonergic compound. So most of the antipsychotics that we're discussing either have predominantly dopaminergic effects, blocking effects, or the atypical antipsychotics, a higher serotonergic 5-HT2A receptor to D2 receptor blocking effects, but they all affect dopamine to some extent. Here you have a pure serotonergic compound that showed antipsychotic benefit and received FDA approval for Parkinson's disease psychosis. I will make one note that we now have an additional antipsychotic that's been approved for schizophrenia that has no dopaminergic, serotonergic effects at all, but only cholinergic effects. So that again raises a different class of compounds and whether that should be considered in the same vein as these other antipsychotics. Next. After the original pimavanserin study, there was another study in dementia-related psychosis called the Harmony study that studied this compound pimavanserin not only in Parkinson's disease, but dementia with Lewy bodies, Alzheimer's disease, and 2 other dementias, showed this kind of robust effect, which was really driven by the Parkinson's disease dementia population. Next slide. But this is really the point I wanted to make, was perhaps compounds that don't have dopaminergic blockade may have less risk for adverse events overall. Here, this just showed good tolerability of this compound in this study. Next. Good tolerability from a motor standpoint using an instrument called the extrapyramidal symptom rating scale. Next. A similar figure for the mini mental state examination score. Next. So DLB is a unique population in its own way. It's related to Parkinson's disease. It's related to Alzheimer's disease. It's kind of an amalgam, but there's great concerns about antipsychotic use in the DLB population. And this comes The quote that I have here from the most recent diagnostic criteria for DLB that were published in 2017. But really all the reports of this extreme antipsychotic sensitivity in this population come from very old literature, older antipsychotics, primarily case reporting. So really very little kind of high quality research to inform this statement. Next. So here are the key takeaways to help set the stage from my standpoint anyway, that psychosis, agitation, and related disorders are common in dementia, Alzheimer's disease, Parkinson's disease, dementia, dementia with Lewy bodies. They often overlap. They're associated with worse outcomes, and that's been clearly established. They often, but not always, but often require treatment. You heard a little bit about non-pharmacologic approaches before. That's really an under-researched area, and most of that research has been done in Alzheimer's disease, but shows that it potentially is helpful for the management of these neuropsychiatric symptoms. Evidence for non-antipsychotic medications are limited. So we're talking about antipsychotics, but other medications such as antidepressants have been used and show some promise for agitation in Alzheimer's disease, but not for psychosis specifically. Cholinesterase inhibitors have been tested for antipsychotic effects and have not clearly shown benefit in any neurodegenerative disease. So antipsychotics themselves appear to be efficacious, but may not be effective when you weigh in the adverse event profile as well. Atypical antipsychotics appear to be an improvement overall in this population. For instance, less likelihood for tardive dyskinesia to develop. But however, there are still concerns about tolerability and morbidity and mortality. Although I will say this is the concluding comment I'll make, from a clinical standpoint, I would say that the elevated risk is there, but it appears to be relatively small. So if you look at 1.6% versus 1%, that gets factored into the decision making. And I would say practically very few clinicians that I work with ever discuss the mortality risk with patients and their care partners. They may discuss other adverse events, but very rarely the mortality risk. Thank you.
Thank you, Daniel. I'm going to ask just one quick question. You mentioned that they are efficacious but not effective, and I'm wondering if you could just come back at a little bit of a higher level for our audience and just explain what those two terms mean.
So in this study, anyway, how that was defined. So if you look at a typical randomized controlled trial for something like this, even, you will have the primary outcome be a measure of psychosis or agitation, and that's your efficacy outcome. All the other reporting is then done separately, looking at adverse events, tolerability, morbidity, could be mortality. And you don't really ever marry those two sets of outcomes in any way. I think the kind of brilliant thing about the KDAD study was it was time to discontinuation as the outcome. And that time to discontinuation could be for any cause, but then you could also look if it was time to discontinuation for lack of efficacy, or it could be time to discontinuation for adverse events. And it turns out when you combine those two into time to discontinuation for any cause, it's a wash. Whatever benefits you have in terms of efficacy are offset by having to discontinue the medication because of adverse events.
So effectiveness is sort of a combination of the two.
Yes, there you go. You summarized it very well.
Wonderful. Thank you so much. Wonderful presentation. At this point, I would like to join the rest of ask the rest of our panelists for this session to join us on the screen. Today, we're joined by a wonderful group of people. Jacobo Minzer, professor in the Department of Health Sciences in the College of Health Professions at the Medical University of South Carolina, and a staff physician at the Ralph H. Johnson VA Healthcare System. We also have Russ Paulson, Chief Operating Officer at USA Against Alzheimer's. And we have Sue Peshin, President and CEO of the Alliance for Aging Research. And finally, we have James Taylor, President and CEO of Voices of Alzheimer's and a former caregiver. Thank you all so much for joining us. I think we're going to go to the next slide, and we're going to see some of the questions that, discussion questions that we're going to be using today to help guide the discussion for this session. I'm not going to go over them quite yet, but we are going to drop them in the chat so that the audience can see what those are. And we're going to be giving our panelists a few minutes for some brief opening remarks before turning to those discussion questions. Jacobo, we're going to start with you.
Well, hi, thank you for inviting me. I want to make clear that although I work for the Medical University and the VA, I'm only sharing here my own personal perceptions. And my overall perception is I am confused. And I'm confused as a clinician because from one type, finally, after lots of years, I have 2 drugs that have indication for specific neuropsychiatric symptoms or neurodegenerative disorders, but at the same time have a black box that tells me it's a bad idea to use them. And my local regulatory people and local authorities are encouraging me to use non-indicated medications that they are off-label because just don't have the black box. As a researcher, I'm very confused because I'm using data from 2005 to evaluate with a big brush a number of compounds with different mechanism of actions that have been studied for different indications and with populations that have been stated clearly that they're heterogeneous. So I'm confused. And finally, I'm confused as a patient advocate because there are treatments for cancer, for example, that have 49% at serious adverse events. And we do it to give to our patients a few more weeks of life with a horrible quality of life. On the other hand, we have here an increase from 1 to 1.7% in a treatment that appears to improve quality of life of individuals, and that happens to be approved by the FDA. So I'm looking forward to understand better the rationale. Thank you.
Thank you. Russ?
Thanks. I didn't know who that man was next to me. Now I know it was Bob, but then I didn't know. Bob was her husband of 60 years, and she had taken a swing at him. It looked like a person. It didn't look like no statue. It looked like a person. And I just took a swing. And if it was a person, I would have laid him out. Another time I pried my eyes open when I see a young woman, a young teenager or whatever. I pried my eyes open and she just kept staring at me. And I thought, I'll be damned. We at Us Against Alzheimer's do a lot of research with patients and we get their stories. And it's common that we say that people with Alzheimer's lack insight into their disease, but not always, not all the time. And I think it's important as we wrestle with these that we realize that these can be very scary things. They can also be sort of interesting. One of the people I quoted later thought that she saw warthogs in the backyard and just thought that was kind of interesting and fun. So they're not always scary, but she knew that this was what was going on. So we talked in the very first presentation of the day, there was a mention about how this results in people being hospitalized, people being put in facilities, which is just a terrible way to talk about a human being. But it's also scary to the people who live with the disease. And so I recognize that the data that the FDA are wrestling with and that have been so thoroughly presented today are really complicated. And I think as a former caregiver myself and a patient advocate, thank you for wrestling so diligently with the data, for taking looks at it from every direction you can. And I think that what some of us on this panel cannot bring medical expertise or statistical expertise, but we can bring the experience of people living with the disease and sort of an understanding of the impact. There's sort of cost and benefit that have to be weighed in all these decisions. And there's a cost of inaction that we have to be aware of as well. Thanks.
Absolutely. Thank you so much. Jim?
Thank you. And I appreciate the opportunity to be here today. I qualify everything I say with no clinical background or training, so I don't speak from the brain. I try to speak from the heart. My wife, Jerry, was diagnosed in 2012 with mild cognitive impairment, later qualified as Alzheimer's disease. She was trained initially as a nurse, moved into hospital administration and research, and ended her career as the CEO of the largest long-term care facility in New York City, having implemented a number of new novel programs. She retired early because she began to have cognitive issues. She was a homozygote and in 2012 was diagnosed. We had quite a remarkable journey with Alzheimer's. We traveled widely and spoke often, encouraging people to live well after being diagnosed with a chronic condition and to participate in clinical trials. And we were very fortunate that Jerry was in the clinical trial of one of the disease-modifying treatments that was later approved by the FDA. Last summer, the summer of 2023, Jerry began to experience symptoms what we call in Alzheimer's sundowning, which I saw listed in the chart of the first presentation. She was aggressive, she was angry, she was delusional. And initially this was occasional and short. As the months progressed, it became almost routine and for multiple hours. It reached the point that I was afraid I would have to call 911 several evenings. I had promised Jerry that she could stay home. She did not want to go to an institution because she wanted to be with the people who loved her and whom she loved. But I was afraid of my wife. I was afraid to keep her at home. And so I didn't feel that I had a choice except to move her to a memory care unit. I think there's a very important point that needs to be called out, and it was been just mentioned previously, and that's that the quality of life is extremely important to the patient and the caregiver. I think we all recognize now that patients and caregivers do not wish to extend life when it comes at a high quality and the quality of life, a high cost in the quality of life. There's a very important parallel, a corollary here. And that's that when the quality of life becomes so impacted, we wish to have the ability to decide on treatment that might have greater risks than we would otherwise consider. And we want to have that choice. We believe it's important for us to be able to say, with a minimal increase in risk, something from 2 to 4% mortality, that should not be precluded from my ability to choose that treatment for my wife, for the person I'm taking care of, for the person I love. Because we don't choose to continue in this condition. It is not a quality of life that we feel should be sustained. The FDA, when they made initial decisions, I saw that the year 2005. I think in 2005, the FDA was not considering the patient voice, the caregiver voice, nearly as much as they are today. The caregiver voice, the patient voice, is give us a choice. Allow us to decide when the quality of life is so diminished that we can take a greater risk than we would have previously. Thank you.
Thank you, Jim. I really appreciate you sharing your lived experience. Sue.
Thank you. Thank you to the Duke-Margolis Institute for Health Policy and to the FDA for convening today's important workshop and for inviting me to speak. There are 3 reasons why we advocated with the American Society of Consultant Pharmacists for this workshop, including a reanalysis of the data behind the FDA's nearly 20-year-old boxed warning on antipsychotics. And the first is to urge the agency to re-examine application of the antipsychotics box warning on a drug-by-drug basis, rather than as a blanket application to the class for both current products and future applications. The FDA's attempt to thread the needle between mortality data on older antipsychotics and newer medications such as Nuplazid and Rexulti, which the agency deems safe and effective, has resulted in overly confusing boxed warning language. And the impact on patients and caregivers is they are not left with good information on the actual risks and benefits of the newer treatment options. The second reason why we advocated for this meeting is to urge the agency to recognize that applying boxed warning to the entire antipsychotic class likely contributes to the stigma of neuropsychiatric symptoms and the prescribing practices of primary care physicians to treat them, including the on-label FDA-approved uses. According to the Alzheimer's Association, about 85% of people with dementia are first diagnosed by a non-dementia specialist, usually primary care physicians. PCPs are also responsible for most of the post-diagnostic care and management of complex needs, like neuropsychiatric symptoms. Alzheimer's and Parkinson's are progressive and fatal diseases. Neuropsychiatric symptoms not only worsen disease-related cognitive and functional decline, but they also accelerate time to institutionalization and eventual death. A large longitudinal observational study that was published in the September 2013 issue of the American Journal of Psychiatry showed that it's the symptoms, not the use of antipsychotic medications, that predicts nursing home admission and death. It's not the FDA's job to tackle stigma, but it is important to understand that application of the box warning may exacerbate it. And that leads me to the third and final reason. While we know it's not the FDA's intent, in practice, the impact of the box warning for people living with Alzheimer's and other neurodegenerative diseases has been, for your sister agency at CMS, to limit access to desperately needed therapies. Since 2020, the Alliance and the ASCP have co-convened Project PAUSE, which stands for Psychoactive Appropriate Use for Safety and Effectiveness. We, alongside members of PAUSE from across the healthcare community, have urged CMS to fix the agency's outdated antipsychotic nursing home quality measures. In fact, a May 2021 HHS IG report that was requested by CMS found that the measures have failed to distinguish between appropriate and inappropriate use. Those measures, buttressed by the box warning, hamper clinicians from following best practices for clinical care, require changes of medication regimens, even for well-managed patients, and they harm the ability of patients' families and their clinicians to appropriately manage care. So we look to all of you. No pressure, but thank you for considering these important issues.
Thank you so much, Sue, and to all of our panelists. I think it's a lot of really great opening thoughts. I do want to remind the audience that we are soliciting questions. Go ahead and if you have them, to put them in the Q&A function in Zoom. We're going to get to as many questions as we can. We are going to start with our discussion questions. So due to time limitations, we may not be able to address everything, but please do go ahead and put those questions in. So I think I want to follow up a little bit on part of Sue's last comments. We're really thinking about how do, and I'm going to break this question into two parts. One is, how do providers communicate the risks and benefits of antipsychotic medications with patients and caregivers when discussing the potential use of management for management of dementia-related behavioral disorders? And maybe I'm going to start with Jim.
I don't know how most providers provide that information. I know for myself, I did not feel that the information was well discussed or provided with me. I felt there was a real hesitation around this topic with the physicians who were taking care of my wife. And I'm sure I can speak for most patients in saying that we do not have a good understanding of the efficacy or the risks with this class of medication. And it needs to be we need to understand it so that we can make a choice and help guide the decisions that are made.
Thank you. Daniel, do you have any thoughts on this?
I had mentioned at the end of my presentation that based on my experience, that I think this information is rarely communicated by most clinicians. I think one, it's because the risk is relatively small, the absolute risk, like 1.6% versus 1%. It's hard to put that into a clinical context. I'd say much more of the time for a clinician, including myself, is spent discussing the more garden variety side effects that I discussed. I also think that the data is a little bit hard to interpret clinically from what we heard before. For instance, most of the studies have enrolled people with relatively low mini-mental state examination scores. We saw an average score of 11, which is really moderate to severe dementia. Many of us are treating patients with less severe dementia, so we wouldn't even know if that risk is the same in patients who are less affected. So there's all these other kinds of variables to consider as well.
Jacopo?
Yes, two questions, two items. First of all, Dan talked about the KD study. Two important things to clarify. The study did not have a clear inclusion criteria in terms of syndrome. The study basically told the physicians, if this is a patient that you will treat with an atypical antipsychotic, then the patient should be in the study, which calls for an extremely heterogeneous group of individuals at different stages of the disease and makes very difficult to move from the average to the individual. The second issue, which is regarding to the communication with the patients, it's rare that a patient will ask me in a situation like this, like agitation, what are the side effects? The patients come and ask the caregivers and the patients when they can express, they said, how can we make this better? Coming back what James said, how can I keep my loved one at home? Would that help? When I tell somebody that have a loved one that is in this extreme situation, the difference between 1 and 1.6% is not something that will be extremely influential between the patient and the caregiver. But there are other elements in the decision making, like for example, the pharmacist, the regulatory authorities, not the FDA, but local regulatory authorities, that will not allow the relation between the patient and the caregiver and the patient and the doctor to be the preeminent decision-making process.
Actually, I think that that's a great segue. I'm gonna be asking the same question, but just a little bit twisted around a slightly different way to Russ and Sue, which is, when we think about the risk information contained in those box warnings and communicated by providers, how does that impact the decision-making process for patients and their caregivers when considering the use of these products? We heard a little bit from Jim, but I'd be interested, and maybe we'll start here with Sue.
I mean, I was looking up over the last couple of days, like just news stories around mention of boxed warnings, not just related to these, but, and there's certainly been a ton of stories on the monoclonal antibody therapies for Alzheimer's disease. And they always say it's it's the most stringent FDA warning that's available. It really, I think, says to the public, these drugs are dangerous. And I think you can't, undercount that. I also think it's important to remember that it's still in the statistics around 50% of cases getting missed in primary care. And some of it is just there's a nihilism, if you don't feel like you have much that you can do, my dad was a doctor, he was a pediatrician for over 40 years. People, clinicians are people, they want to feel like they can do something for their patients. And, again, I think this gets into, I really respect the FDA. We are been part of the Alliance for a Stronger FDA for so long. We love the agency. Please kind of understand this in context that, you want, there are very few options here for folks. And, the use of the sort of leveraging of these warnings by the payer community is, I think, an important implication that the agency needs to take into consideration, just like they do for REMS or other types of warnings. And that really is what, concerns us, because it filters down to the patient community. And we're so excited that there are 2 medications that have been approved so far. We hope that there's more options to come. We don't believe in chemical restraint. These are not appropriate for everybody. But, it's like pretty amazing to just have options. And, so that's why we're asking for reconsideration of the newer medications, because we feel like it's based on this old data and it's kind of out of context. So that's my response. Thank you.
Thanks,
I
Sue.
think I would say, Christina, to your question, it really does depend on how the physician presents the information. Docs can present it as, it's a box warning on your ibuprofen as well, and sort of minimize the consequences of the box warning. Or it can be, oh my God, it's a box warning. There's a lot of, in the Alzheimer's world, especially, I don't know how it is in Parkinson's, Dr. Weintraub, but in Alzheimer's, there's a lot of variability in how clinicians think about these drugs and this illness in particular. So it really matters a lot how the doc presents it. That said, you heard Jacobo and Jim both say people want to make this better. And to the my opening sort of anecdotes, these are scary things for the patient. They're scary for the caregiver. And whether it's a risk of mortality or morbidity, people are very interested in making the thing in front of them, which is severely debilitating, severely affecting quality of life, may cause someone to go live someplace else, which is what a lot of people want to avoid at almost any cost. They'll go pretty far and sort of discount, unless the doc says, oh my God, this thing has a black box warning. This is the strictest warning the FDA can give, blah, blah, blah. So it does a lot depend on, and that's too much in Alzheimer's, it depends on who your doctor is.
I would like to add one important thing. I congratulate the FDA for having the first 2 drugs approved for these neuropsychiatric symptoms of dementia. That somebody that has been in the trenches for 30 years or more, I'm just disclosing how old I am, but that's okay, that it was a moment of joy. But I think it's important then to increase the chances that these drugs will be used by providing information that it comes from the clinical trials that led to the approval of the drug, rather than an analysis that was done in 2005.
Christina, if I could just add one point.
Please do.
We all want more disease-modifying therapies, and I feel that Jerry and I benefited from one. But now, having completed my journey with Alzheimer's, disease-modifying therapies give us longer life. But if there's one thing I would ask for first, it would be effective antipsychotic drugs, because that effectively was death. It wasn't worth continuing when Jerry reached the point that I had to put her in a home where she was never happy, and I visited every day and didn't see many happy people there, neither the care partners nor the patients. And the current stigma around antipsychotics is limiting pharma investment and research into these drugs, which are to me, that's where the greatest need is. So we need to come up with effective labeling that says there is a risk here, consider it, but not prevent it from being in the hands of effective physicians and giving patients and care partners the choice to use the medication.
Thank you, Jim. And I think that actually dovetails really well into our next question, which is actually about those considerations. What considerations might providers weigh when assessing the utility and appropriateness of these antipsychotics for treatment of dementia-related behavioral disorders? And I think we're going to start with Jacobo and Dan, and then I think that I'd like to also hear what patients and caregivers think that should be weighed in there. So if we can start with you, Jacobo.
we need tools to treat our patients. And again, thank you, agency, for giving us some tools that we didn't have before. But when these tools needs to come in the right reference, physicians also in general are adverse to risk. The presence of a black box increases the risk of the physician to prescribe a certain medication. And not to mention in long-term care, where I have a drug that is indicated, that is approved by the FDA, that helps the patient, but I need to discontinue after 30 days because of regulations. So it's one of the few cases in medicine where I have a treatment that is effective and useful and approved, and I need to stop it. So there are many factors, and I think the black box goes way beyond a warning. But I have lots of effects that I'm sure were not intended and may not be in the scope of the FDA to decide, but they are real in the real world.
Dan, do you have anything you wanted to add?
Maybe just that if I understood your question correctly, I think I really rely on the severity of symptoms kind of in a broad sense to guide treatment. So that may be from the patient reporting how frequent they are, how upsetting the symptoms are, how disruptive they are to things like sleep, and then of course involving the care partner, caregiver in terms of impact on that person or the home environment as well, because that's often what's driving the bad outcomes, or if there's long-term placement involved. And then, constantly like reevaluating. I think the one good thing that's come out of all of this is I think we've tried to learn about using non-pharmacologic treatments. We've learned to make sure we're not missing some medical conditions such as urinary tract infection. I think we're better at reevaluating treatment over time, like not just putting somebody on antipsychotic and then forgetting about them, but is there an ongoing need, or can there be trials off of the medication considered? So I think those are some of the other factors at the clinical level.
Thank you. Russ, do you have anything you'd like to chat about on this question of really, when providers are weighing considerations around assessing the utility and appropriateness, what would you want them to be considering?
What I want them to be considering. I was going to say I actually agree with everything that Jacobo and Dan said, but I would add, I guess, and I said it a couple times, I'll say it again, there is a cost of non-action as well, and I think docs do that automatically, but I don't know, to Sue's point earlier, I'm not sure that payers always do, but we would hope docs would think about the severity. And Dan talked about the severity of the symptoms. The presence of the symptom in and of itself is pretty severe. Like it is just seeing your loved one hallucinate, or knowing that you yourself are hallucinating, is in and of itself a pretty big deal. Even if it's just, I think I see a warthog in the backyard, versus I think that there's a strange man in my bedroom and I need to punch him. It's still scary.
Yeah.
Jacobo?
I want to bring two patients for just a couple of seconds. One of my dearest colleagues have Parkinson's disease, and he's self-diagnosed, and he's psychotic. And he told me, Jacob, this is terrible. And I said, why? I know the images are not real. I know somebody doesn't try to hurt me, but I cannot stop running. The treatment release is something that improve or decrease his suffering. The second is a patient with Alzheimer's disease, not so severe as those in the studies, that came to me and said, Doc, I know I'm not reacting well. I know I make my loved ones suffering when I react like this and get so upset and agitated. I just cannot stop it. Can you help me to stop it? So we have to bring the patient into the conversation and see the personal suffering that the individual has, and find a way to alleviate, because that's what we do as physicians. And agree with Dan, after we are sure that there are no medical conditions, that we try behavioral interventions and all of that. But when every tool has gone, we need to intervene pharmacologically.
Mm-hmm.
Did you have anything you wanted to add to that?
Did you ask me?
Yes, I'm sorry, Sue.
Oh, sorry. I agree with comments that have been made before. I mean, I think that it's wonderful, you know, that the FDA did approve 2 medications, and I think that's actually resulted in advocacy organizations and provider groups creating a lot more resources that clinicians can use and long-term care facilities can use around management of neuropsychiatric symptoms, and like Dr. Weintraub said, you know, identifying any kind of underlying medical issues first, and then trying non-pharm. And you know, my mom sort of taught me validation when my great grandmother had what was then called senility years ago, when she had psychosis, and we would just sort of go with it. And there's so much that can be done through that. But you asked about the question of what do you want providers to consider, and I think dignity is a big issue, you know, of just because, you know, people lose control of, you know, their ability to go to the bathroom when they have extreme NPS. It's not, you know, it's rough, and it can be it can be safety related for the individual as well as the caregivers. And I think all those things should be taken into consideration. And I also just want to put out there, I don't, you know, think families make these decisions lightly. I also don't think people in long-term care facilities make them lightly either. I think sometimes there is a perception that we just throw a drug at it. And maybe that does happen in some inappropriate situations, and we would be, you know, opposed to that. But just, you know, people want relief, and I think that there's just an opportunity here to really address stigma by reopening these issues. So thanks.
Thank you. And Jim, we started this question actually jumping off some comments you had ended with, but I wanted to give you a chance if you wanted to add anything else. And you're muted. I'm sorry, Jim, you're still muted. There you are.
I have nothing to add. I agree with all the wisdom that was expressed. And I think, you know, just in this panel, by including the patient experience, by including the caregiver experience, people want to know, people want to hear from us. And that's very encouraging. It gives our community great hope. And we trust that what we say is going to be weighed, is going to be listened to, and that the best decisions will prevail.
Thank you. I'm going to turn to our third discussion question, and I just want to remind the audience, if you do have questions, please go ahead and put them in the Q&A. We want to make sure that we are getting to audience thoughts and questions as well. And so I want to move a little bit to thinking about research, and asking sort of what research questions may be needed to may need to be addressed to best inform patients, families, and clinicians of the key considerations and appropriate use of these antipsychotics for this patient population. I think I'm going to start with Jacobo here too.
Yes, I think that our colleagues from the FDA frame it extremely well. And I think that we need to move from the average to the individual. In other words, we should be able to tell that an individual at a certain level of cognitive impairment with these type of comorbidities has a risk of blank, I'll taking it, and then allows people to make an informed decision. I think the data we have now, not meant to be dismissal, but it's not meaningful because there is too much noise to signal and too much heterogeneity that doesn't allow me to use that data to make an informed decision to the individual I have in front of me.
Yeah. So you maybe hit a little bit on this, and I want to give you a chance to expand a little bit when you talked about separating out the medications and looking at them individually. I didn't know if you wanted to add on anything there.
But the mechanism of, if we assume that all the antipsychotics, and we define antipsychotic for what they do, not for how they do it, right? We're defining because they improve psychosis. It's like to say that everybody can drive is the same, right? And then make a decision globally. A better way would be to define things by the mechanism of action that we think is involved in the adverse events or the side effects or the mortality that we observed, rather than to say, if you have this class, that's what it is. And I think that that's the main problem of the analysis that was done in 2005. Plus, there is some data today that really won't support it. The study I have the pleasure that Daniel presented in the beginning by Dave Devanand, where we were very careful in the kind of individuals that we were selecting. And we were careful in evaluating the risk of cardiovascular disorders that these individuals. Plus, we're careful about the indication. We didn't see any signal. The signal wasn't there. So I don't need to tell my colleagues that if you use the wrong medication for the wrong patient, you will see lots of side effects. And probably that's a bad idea.
Daniel, do you have anything you wanted to add to this discussion?
I like Jacobo's points. I think some studies are hard to do. Like some questions can only be answered with very longer term studies. As was already mentioned, all of these studies are acute studies we're referring to, which are informative, but don't tell the whole story. Long-term studies are hard to conduct, they're costly to conduct. I think one thing that was nice about the KD-AD study was it was like active comparator studies, or so platform type trials. I think being able to compare antipsychotics in some way would be helpful, informative for clinicians. We have very little data about that. And then finally, I do like the idea of, in the shorter term, looking at the predictive factors. So clinicians are informed about who might be an increased risk. Is it more severe dementia, some other characteristic of the patient? I think now that we're moving more to biological characterization of patients, knowing people that are, for instance, biomarker positive for Alzheimer's disease or biomarker positive for synuclein disease really lets us know who we're dealing with, as opposed to just these kinds of somewhat inaccurate clinical diagnoses we use.
Thank you. Sue.
Yeah, I'm sort of curious about why the FDA decided not to include a boxed warning on Cobenfy, the antipsychotic that was recently approved by the FDA to treat schizophrenia in adults. One thing I was, thinking about was the average age of schizophrenia onset tends to be much younger. It's in the late teens and early 20s for men and late 20s to early 30s for women. The average age of onset for Alzheimer's disease is 65 or older. So it'd be helpful to understand how the agency looks at mortality by age group in different disease populations. And just sort of overall, I think what the FDA considers the rules of the road for applying the boxed warning. In relation to the previous question that we talked about, that made big news when the drug was approved, that there wasn't a boxed warning. And so it does sort of set up this difference. It sort of contributes to points of view on the various medications. So that's of interest from our perspective. I don't know if it's so much like research on behalf of the FDA, but I just think it would be good to, have a bit more sort of transparency around all of that.
Thank you. Jacob, I see you put your hand back up.
Yeah, I would like to support the comment that the FDA made in terms of using blood-based biomarkers and other biomarkers. The amount of people that look like Alzheimer's disease but will be negative in biomarkers is give or take 30%. Mm-hmm. These 30% can clearly change the side effect profile one way or the other. So I think that we shouldn't be labeling things in the 21st century with technology and information of the 20th century. And I think we need to really update to be able to provide accurate information for our patients.
And I do see that we got one. We do have an audience question, so I'm going to read this. One audience member commented that the diversity of causes that explain the increased mortality risk suggest a lack of a direct mechanism. This raises the possibility that use of these products may impact the pattern of care. From a clinician perspective, so I'm going to be asking Jacobo and Daniel, might the use of these drugs impact prompt recognition of acute illness and otherwise facilitate missing care needs? And would this problem also result with the use of other drugs?
I always start, so Daniel, you start first. I
would just say it is confusing and not known. So if you look at the morbidity risk for antipsychotics and dementia, it's related to cerebrovascular events. If you look at the mortality risk, it seems to be somehow related maybe to cardiovascular events or pneumonia. So I don't really think we know what is driving the increased risk. And I agree, it's very muddy waters at this point.
Just to add to that is just to reiterate that it's the obligation of the clinician to evaluate each individual patient's risk. And it's committed to the physician to rule out medical problems such as cardiovascular disease, stroke, pain, that may be affecting to the patient and the patient cannot appropriately express. But when we rule all these out, then that's when we need to know the effect, and we have the exact definition, and we have the exact profile of the patient. That's when we can assess the risk of taking a certain medication.
Thank you. We have 2 minutes left in this session, so I am going to move to closing remarks. Any last thoughts that you may have? Russ, I'm going to start with you.
So quickly, to the last question before the question from the audience that you asked, what research is needed? We've done a lot of research, and even at the earliest stages of the disease, people with pre-symptomatic disease but biological risk, people worry about these neuropsychiatric symptoms. This is an important topic for people even before they start manifesting the symptoms. So we know that it's important. I think what patients would like to know, and if the research currently doesn't exist, and maybe the individual level data gives you this, what's my chance of benefiting from this? What's my chance of having a risk from this? And what does benefiting look like? How, and just know patients think about it in terms of time. So how long is it between episodes? Will you prolong the interval between episodes? Is that, or will you reduce the number in a way, which basically says the same thing, right? So I think you've got a lot of that data already, but there's probably more. And I'll just close by saying again what I said at the beginning, having heard all of the data that FDA is looking at, I don't envy the job. I'm very glad as a caregiver, as an American, that we have such dedicated people at the FDA wrestling with all of the uncertainty that's attendant to this. And we as patients stand ready to provide any kind of perspectives we can to help in that analysis. So thank you for having patients and patient advocates and caregivers here today. And thanks for wrestling with this important topic.
Thank you. Jacobo, any last thoughts?
Yes. I was born in Argentina from a mother from Syrian origin and a father from Poland. But I came to the US, I discovered that I'm Hispanic. Now, I don't think it's a good idea to generalize things for phenotypes that may not be in common. And therefore, if somebody comes and evaluates my risk for any medication, well, I probably would not be a good risk factor to think about for me for being Hispanic. So I would suggest that we need to move, as I said before, from the average to the individual, so we can understand what is the risk benefit for at least a quite distinct group of patients.
Thank you. Sue.
Want to reiterate for the agency to evaluate the drugs on a drug by drug basis, and really just to thank you again for doing this. It's like hitting a wall at your sister agency's side of things. So it's just very refreshing to have this kind of dialogue with all of you. And I want to reiterate what Dr. Mincer said about just appreciating your service and the emphasis on safety and public health. Thank you for everything that you do.
Thank you, Sue. Dan?
So I would say that psychosis and agitation across neurodegenerative diseases is a big problem. It's still, I would say, a big unmet need because we do have some treatments available, but clearly not enough. And I do think there's evidence that antipsychotics overall can be efficacious in Alzheimer's, Parkinson's disease, even dementia with Lewy bodies. And is there an increased risk? Maybe. I mean, I wouldn't even say that that's 100% clear because it's only when you look at aggregated data that it really becomes more clear, not at the individual clinical trial data as much. So I can imagine there being a warning, but I don't know that there needs to be a black box warning still.
Thank you. And Jim, I'm going to give you the last word.
It's an honor for me to be included with the people on this panel today and for me to be here. So thank you for honoring patients and caregivers in that way. You know, we can scrub all the data and maybe we can answer some of the questions that Russ raised that would be nice to know. But, you know, in the end, it's always a matter of choice. It's a matter of faith. We won't ever know everything. And we can only, for each individual, say what the impact is on my life, on the life of the person I'm caring for. So again, by being able to make this choice and understanding this choice, which is the job of the folks on this panel, and in communicating that, I believe, can we really help the patient who really is our ultimate customer here. So I just am grateful for the progress we're making, and I'm very appreciative for this panel today. Thank you.
Thank you so much. And I just want to share our incredible gratitude for the panel for talking to us today about your work and your, like I said, your lived experiences on this topic. I really appreciate it. We're now going to turn back to Nancy for our final session of the day. Nancy?
Great. Thank you, Christina, and for that whole panel. That was a absolutely wonderful panel, and I think has teed us up really well, highlighting the complexities, the heterogeneity, the challenges that are faced for this last panel of the day. So in this final session, we're going to cover opportunities for further characterizing value and need for box warnings for antipsychotics. During this session, we're going to have participants will consider what outstanding clinical and scientific questions may need to be addressed through future efforts to better understand the mortality risk with the use of antipsychotics in individuals with behavioral and psychologic symptoms of dementia, including teasing out a better understanding of risk modifiers. Before we turn to the full panel, I want to welcome back Shamir Kalaria, who will be providing a brief presentation to get us started. Shamir, go ahead and take it away.
Thanks, Nancy. And I just want to say that was such a great discussion to listen to, and just looking at the caregiver and patient's perspective. And I think this is a great segue into kind of some action items that we can undertake as an agency to move forward with this signal. Nancy, could you progress to the next slide, please? So to preface this panel discussion, I do want to acknowledge, again, I know we discussed this at length, but the limitations of the available subject level data that we have from a few programs that Fengyu had mentioned earlier. The first, and I think the most important, is, again, all these clinical studies are limited to only short-term data. The typical study duration here is about 10 weeks in length, with the largest study being 26 weeks. So we do acknowledge that there are open label safety studies, but the interpretation of those studies may be complicated by the open label nature of those studies, and then also the lack of a control arm. When looking at the patient level data, specifically in terms of patient information, there is medical history that's reported, but may be unreliable. This is based off of the practice of running clinical trials back in the 1990s, where some of the reported self-report patient history may not be reliable. EHR data may not be as reliable to understand specifically the patients that are being enrolled in the studies. And this also alludes to the limited information we have on prior and concomitant medications, whether a patient was taking a specific medication that could be at risk, were they receiving antipsychotics prior to enrolling in this study, were there other treatments such as benzodiazepines that were administered to these patients during this study. We really don't have that information from some of the older trials. And then ultimately at the end, as a lot of the panelists in the previous session alluded to, the studies are extremely heterogeneous in terms of the patient populations. There were different diagnostic criteria, especially the clinical criteria in defining Alzheimer's disease and also the NPS symptoms. Again, lack of biological definitions for the dementia subtypes. And then we also acknowledge that there are a lot of co-occurring NPS symptoms that are occurring in this patient population as well. So how do we tease out the specific symptoms, changes in the symptoms as it relates to the increased risk? Next slide, please. So in terms of future steps, we're planning to do a comprehensive analysis of the questions and discussion items from this workshop to inform the need for additional data and analyses. And as Feng Yu and others have mentioned this before, we have pending responses from sponsors to conduct subjects-level analysis from available randomized controlled trials. And this includes data from the brexpiprazole and pimavanserin programs as well. And then obviously, we do understand there's a lot of information in the current literature, including observational studies. So is there a potential to conduct a meta-analysis of the available observational studies to help complement the subject-level analyses that are submitted from randomized controlled trials? And the last slide, please. So this brings up to some of the questions that will lead to our discussion for the panel. The first question is, how might the agency consider risk modifiers, such as the type of neuropsychiatric symptoms or the type of dementia? Can risk modifiers related to medical comorbidities be realistically accounted for in the analysis? And what would be some adequate research methodologies to account for these medical comorbidities and changes in the symptoms as it relates to efficacy? And then if a new product with indication for the treatment of neuropsychiatric symptoms of dementia is submitted to the agency, how might the agency evaluate whether there is an associated increased risk in mortality, and if relevant, contextualize that risk? And that kind of goes back to the question about, do we take this on a case-by-case basis, the generalizability of the box warning. And so these are some of the questions that we have brought up as an agency to consider further for this signal. And I will turn it over to Nancy and the rest of the panel to provide their insights and their expertise to help facilitate this discussion. Thank you.
Okay, great. I think my camera's having a little trouble right now, but hopefully it will behave itself. I wanted to bring up all the members of the panel, and I'm going to go through and introduce each individual, and then I will ask each of you to give a couple of minutes of opening remarks before we get into the discussion questions. So first on this panel, we have Rebecca Edelmeyer, who's Vice President, Scientific Engagement for the Alzheimer's Association. Valentina Mantua, who is a psychiatrist serving as Clinical Team Leader at the Division of Psychiatry in CEDAR at the FDA. We have Christina McLinden with the Clinical Interventions and Diagnostics branches at the National Institute on Aging. We have Paul Rosenberg, Professor of Psychiatry and Behavioral Sciences at Johns Hopkins University School of Medicine. Matt Sokup, who's Deputy Director of the Division of Biometrics 7 at the FDA. And Chad Wars, Chief Executive of the American Society of Consultant Pharmacists. So thank all of you for joining us. And I'm going to go through and just ask each of you to provide a couple of minutes of opening remarks. It could be reflecting upon things you've heard today. It could be sort of your high-level area of interest or where you think the most emphasis needs to be placed moving forward. And I'm going to actually start with Paul. Paul, go ahead.
First of all, is my sound okay?
Yeah, sounds great.
Because I was on a prior Zoom call and it was awful. Okay, I'm a psychiatrist who does Alzheimer's trials and treats neuropsychiatric symptoms of Alzheimer's. I not only thank the panel for the opportunity, but I thank the FDA for doing a fresh meta-analysis and some fresh observational meta studies. It's reassuring the numbers are not different from the ones we've been using for 20 years. And it's reassuring to know that in general, there's a lot of evidence for mortality risk. It's not completely consistent. And the magnitude of that risk however you want to measure it, isn't that very different from what we observed 20 years ago. I've been talking to families about this for 20 years when I prescribe it, when I choose to prescribe antipsychotics. And I would say over 90% of families hear about the magnitude of the mortality risk, and they don't exactly yawn, but they basically say, well, that doesn't sound like so much. And in my opinion, as a clinician, these risks are real, but not very large, and they need to be weighed against family and patient distress. There's a lot of distress out there, and I'm generally using antipsychotics not so early in disease, at a point where you're really thinking more about comfort care than you're thinking more about quality of life than quantity of life. And in the context of comfort care, the risks are relatively small. We have a brave new world out here of biomarkers and diagnosis, and we should take full advantage of it. We should really, now that we can define Parkinson's for real and DLB for real and Alzheimer's for real, now that we can do these, now that we have biomarkers and in many cases blood-based biomarkers, we should ask the question how these drugs work in biomarker-proven disease. It may not be different, and it may not be related to diagnosis at all. It may be transdiagnostic, but I think we should start with sharply defined diagnoses. We won't be able to get to diagnostic unless we can get to good diagnosis. I think the main factor here is acuity, and I want to give the analogy of ECT. ECT works great and is safe enough if you restrict its use to severe treatment refractory depression or psychotic depression. The safety hasn't changed from mild depression, but the risk-benefit is very different in severe depression. I think we're talking, one of the reasons I'm gratified to see representatives of patients and stakeholder groups is in this case, we not only need patient-reported outcomes, although many of our patients cannot report an outcome very validly, but they can say whether they feel better, but also, I don't know if we use this much, CROs, caregiver-reported outcomes. And the last thing in my clinical practice, I think we have to distinguish psychosis and agitation. Once again, just like with transdiagnostic, they might lump together, but in the clinical world, still the two are quite distinguishable. Anyway, that's my 2 cents, and I'm sticking with it.
Okay.
Or maybe not.
Great. Thanks, Paul. I'm going to go to Rebecca next.
Hi, everyone. As Nancy noted, I work for the Alzheimer's Association. I'm a neuroscientist with the medical and scientific relations team here. And I think as all of you have really heard already, that many people find the changes in behavior as well as the neuropsychiatric events caused by Alzheimer's and other dementia to be some of the most challenging and distressing effects of these diseases. And I think the association certainly encourages really non-drug approaches as first-line approaches to addressing these features of disease. But obviously, if those are failing after being applied consistently, introducing medications are certainly an appropriate option for individuals living with severe symptoms, and particularly those that have risk or potential to harm themselves or others. So I think that is important to really think about even as we're discussing this, is while many medications can certainly be effective, they also have to be carefully used, whether they have a boxed warning or not a boxed warning, and really combined with non-drug approaches. But I think it's really important, as I've been listening to the conversations today, for the FDA to consider the patients, the caregivers, and that clinicians have already given a lot of thought to how best to manage these really challenging symptoms. And when thinking about the risk benefit, many individuals are already literally at their wits' end when trying to find a solution to improve the quality of life of their loved one or for themselves. And it's critical that there's options available for patients and families. One of the reasons why the FDA is certainly a big part of this role is making sure those options are available. But the options need to be backed by data and supporting evidence to help support prescribing. So I know that even though today we've heard a lot about the limitations to some of the data that might be available to support answering all the questions we have around value and need, I'm really looking forward to also making some suggestions as part of this panel on how we might be able to support that data collection.
Okay, terrific. Thank you, Rebecca. I'm going to turn to Chad next.
Thank you. First, I'd like to thank the Duke-Margolis Institute for convening this call today and the FDA for putting this together. I'm Chad Wars. I'm the Chief Executive for the American Society of Consultant Pharmacists. I'm a long-term care consultant pharmacist by background. And my 2 to 3 minutes is really about the challenges that are faced on the front lines with the prescribing and the management of medications once it is determined that they are required for a particular indication. And I think a number of people on the prior panels and certainly my co-panelists on this panel have brought up some of the key issues. We look to the FDA for, and the public looks to the FDA, and government agencies look to the FDA for truth about the safety and efficacy of these medications. And for the past 10 years in long-term care settings, the box warning has presented somewhat of a barrier. And the implication of a warning across a drug class that treats specifically serious mental illness, in particular psychosis of differing forms, is that the treatment is actually associated with an increase in mortality. And that's a challenge from a prescriber perspective and from a clinician perspective. A number of the panelists have talked about what matters to patients, what matters to caregivers when it comes to dealing with these serious symptoms and potential opportunities to have medications that can alleviate those symptoms or provide some increased quality of life to individuals if they get to the right medication. And I think that does have to be paramount when we consider the impact of something like a box warning on a medication. Certainly there's room for box warnings. We need box warnings as clinicians to help guide our decision-making, but those box warnings need to be specific to the medications that they're applied to. And the blanket application based on a drug class has limited ability. And I think it's, by nature of some of the people, again, on the panel, it's created some challenges for us. We know in particular that clinicians shy away from antipsychotics for all indications because of the warnings, because of the rhetoric around the increased risk of mortality, and because it doesn't present in plain speak. It doesn't tell you what the risk-benefit ratio is. It results in people choosing other medications, other dangerous medications that may have less evidence, less data, less safety implications to be used for some of those symptoms. And that just puts us further and further behind getting patients good treatment. So I think from our perspective as pharmacists that are in skilled nursing facilities on a monthly basis, working with the medical teams to help manage these medications, we look for clarity from the FDA, and we want to understand the process that they went to apply a warning to a particular medication, and it's that based on that medication's individual characteristics in terms of safety and efficacy, so that we can in turn apply it appropriately to the patients that we take care of. Thanks.
Great, thanks, Chad. I'll turn to Christina next.
Sure. I just want to echo thanks to the FDA and the Duke-Margolis Institute for their responsiveness in holding this important discussion. So I think it's critical that we continue supporting research to identify novel, effective interventions for psychosis. It's important that we have a host of interventions at our disposal. As stated by so many others, non-pharmacological intervention should be the first line of care. And I think it's important that in addition to UTIs and other contributing factors, that it's important to rule out hearing or vision impairments, which can often contribute to psychosis. But among those who don't respond to behavioral interventions, then we do need to look to drugs. But I think we should also consider devices as well. We may be able to leverage additive effects through combination approaches between farm-farm and farm-non-farm interventions. Many trials are structured around a standard 12 weeks of intervention, but psychotic symptoms may resolve naturally during that time. So this may be one of the few areas where a trial of shorter duration may be preferred. Perhaps 6 to 8 weeks would be more appropriate. And finally, as was made clear by our stakeholders in the previous session, the care and caregiving burden, distress, and patient quality of life cannot be overlooked and need to be included as key outcomes in all NPS trials. Thanks.
Great, thanks, Christina. I'm going to turn to Valentina next.
Hello. I introduced myself already. I'm Valentina Mantua. I'm a clinical team leader in the Division of Psychiatry. And I would say on behalf of the agency, I would like to emphasize a message that we would like to deliver to the public, that this workshop is indeed a first step towards the revision of the box warning, which we consider being a process that needs to be informed by data. In addition to being informed by data, it will be informed by suggestions, insight, even criticism that was raised during this workshop. Everything will be taken into consideration, and I will try to address some of these insights during this panel. The content of this workshop, as well as data, will inform future steps. Now, we already know what some of those future steps will be, some we don't yet. Certainly, we will, as you've heard from Dr. Calaria before, we are awaiting relevant sponsors to submit subject level data from these clinical trials. These will contain some outcome related information that will allow maybe, however, we don't know, some subgroup analysis that will inform some of the important questions that you've made that we also have. Some of these questions are: what is the relative contribution of the type of neuropsychiatric symptoms? Is it psychosis or agitation? Do psychosis or agitation have a different type of risk? And does it matter how long they've been lingering around, how severe they are? All these questions are still open. Also, other questions are: what is the relative contribution of the underlying pathology of the dementia diagnosis? Now, these type of questions we already know will not be answered by clinical trials from 2005, because at that time there was not that definition. Another important question is: what is the role of medical comorbidities? This is a population, you've seen the population from the meta-analysis as an average age of 80 years. And they have multiple comorbidities. And when we looked at the narratives of each single case of death, for ourselves as physicians, was complicated to disentangle this chain of events that brought to death, to understand what is the unique cause of diagnosis for most of these cases. So there's that, and there's no optimal way of determining that if this is what we've been submitted. As you know, there will be other things that we'll be looking at in the future. Now, two things have been said: that we've approved 2 drugs for this indication. And I appreciate Sue from Alliance for Aging comment that these are overly complicated box languages. And the reason why that is because they describe exactly what's in there. It's like a picture. However, we do understand that we need the bigger picture. We need to put this in context. And the only way that we can do that is to look at data of modern clinical trials. So another question that was raised is: why didn't you include the data from the recent trials? Well, it's hardly comparable. And this is, again, a first step. And modern clinical trials have different quality standards compared to old clinical trials. So we are building our new data set out of our new data. And there's a few things to say about this. Now, first thing I said, the quality standards. And then the other one is that the population's better defined. If there's Alzheimer's disease, most likely, maybe not in the Brex Peepersol data, but most likely in future clinical trials, we will have biomarkers for defining the pathology. We will have a more homogeneous population, and that's gonna shed new light into what is the relative contribution of the actual, pathology to the risk. Another very important thing is as there are new drugs in development, these new drugs may have new mechanisms of action. And that is another point that was raised. Why didn't you apply the box warning to the new antipsychotic that you've approved? Now, first of all, this is a different indication. Second of all, this is a new mechanism of action. And third, the population was not even over 65, so it was not representative. So we do look at data, and we will look at data as they come in, because there are several products that are in development. We will be able to learn whether new mechanisms of action carry the same risk or not, and that will bring us back to the original question. So one thing that I am guessing, and I probably know at this point, that the public would want to know from the FDA is what approach you will have once a new drug comes in for approval. For this indication, not another indication, for this population, what is your approach? Well, I want to emphasize that our approach is that of looking at data. And if the box warning was applied to the pimavanserin and brexpiprazole label, was because there was imbalances in deaths. And so we will look at that. And I also want to emphasize that we always balance, whenever we take a regulatory action that is informative for safety, what are the implications for access? This is very important whenever we take any decision, including RAMs. So I guess I want to say that this is again, a process. And we will make a determination on a case-by-case basis for new drugs. But we hope that we will have new learnings as we proceed with our data analysis and our process.
Great. Thanks, Valentina. You answered a lot of the questions that came up in the prior session. So appreciate that. I'm going to turn to our last panelist, Matt.
Thanks, everyone. And I've really enjoyed today. So thanks, everyone, for their feedback. I wanted to, rewind a little bit and initially kind of talk the reason why there was a meta-analysis even done. And the reason being is you start seeing imbalances on a very serious risk, which here is mortality, which is the ultimate risk. And you start seeing imbalances. It could be within a drug, or maybe starting to see it across. And you realize you don't have enough information within a single product for how it was developed to really have what you believe is a reliable estimate of that risk. So that's many of the reasons why we then go ahead and conduct a meta-analysis. And realizing meta-analyses are only as good as the quality of the trials that ultimately generate the data. But the reason why we find that useful from a regulatory perspective is that estimate we could get from the meta-analysis, if we realize that we have enough trial information to get somewhat reliable estimate of risk, it informs our public health decisions. And that's a lot of what we have to do, is we have to make decisions, on a population level. It's not on an individual level. And we realize benefit risk decisions from an individual perspective would be very different than maybe a benefit risk decision on a population level. And seeing what we saw, which is an imbalance in mortality from the trials and the set of drugs that were utilized in it, it raises a lot of questions, because anytime we see a risk, the next questions are, well, is there someone less at risk, or can we mitigate this risk somehow? Is it dose related? We saw a lot of great questions from our participants today that asked those, and those are all the questions we asked too. But as Shamir and Fengyu have pointed out, is the database to support those questions, to answer them what we feel is reliably, is we don't know yet. We've asked for the data. I have some level of optimism we might be able to answer some questions, but there are likely to be questions we still won't be able to answer that do require patient level information, because that ultimately in many ways depends upon the conduct and design of the component trials that were utilized in the meta-analysis. But again, I think it's something that is worthwhile to go back and expend some resources to look at, because it's really not cost prohibitive. It's really more resource intensive, and I think it's worth putting resources into that. What I think is the biggest opportunity is moving forward, and really thinking about all these uncertainties that we've heard about, and designing clinical programs to evaluate both benefits and risks that then can be utilized to make real public health decisions, and even to help patient prescribers make the decisions themselves. And that, I think we've heard a lot of great information, as Valentina has said, And, statistically, we can support those, if we get the right clinical questions, and we're hearing a lot of great feedback, we can then utilize proper statistical methods to really get reliable estimates and get ideas of benefits and risks with estimates that can inform decisions both at a population level and a patient level. And I'll stop there. Thank you.
Great. Thanks, Matt, for those opening remarks. I'd also like to remind the audience, please continue to submit your questions. We will try to get to as many of those as possible during the remaining time we have. But even if we don't get to your question live on this, it is still very helpful for the panel and the group to hear it and see it later, and they will be compiled later. Now let's get to the discussion questions that we had actually. I think Shamir had presented these in his end of his presentation as well. And I'm going to go ahead and start with the first one here. I know we've touched on a lot of this already. The folks in the prior session touched on some of these issues as well, but I wanted to kind of get maybe diving in a little bit deeper from each person's perspective here of how might the agency consider risk modifiers such as type of neuropsychiatric symptoms or type of dementia. And I know, you can even throw other things in there that you've heard today, that you've gleaned from what folks have said today that might be an important risk modifier to incorporate, and how best to consider that in the context. And we're going to get to some more questions about contextualizing all of this later, but I wanted to kind of start with this level. First, who would like to take a stab at that first? Paul, I see you came off mute, but I don't know. Would you like to go ahead? Okay.
Sure. I talk too much, so I'll try not to. I think this is a great opportunity and to look at a number of, dichotomous or categorical modifiers. They're not that hard to define. In terms of type of dementia, it's great. As I said before, it'd be great if we had a drug that worked transdiagnostically, but that's still to be proven. I think you're best off if you could stratify by dementia diagnosis. Alzheimer's, if you have blood, now we can really make decent diagnoses. And if pharma can be persuaded, I am confident that the makers of pimavanserin and brexpiprazole have samples still in their freezers and may have even analyzed already for p-tau. It's harder to define vascular dementia. It really is. In terms of other stratifications, acuity. The problem is that you've got different measures, but you can find some way with z-scores or something to harmonize the different measures. The reason I'm saying this, not to brag, I'm just talking here. I've written innumerable articles and grants about the treatment of agitation, and some of you have had the bad luck to actually read them. And every one of them has the same first paragraph, that the first treatment for agitation is always non-pharmacologic, Christina, is always non-pharmacologic. And we usually quote the DICE guidelines, which are particularly useful in ruling out delirium. I think antipsychotics probably have more of a clinical place in more severe dementias. I really do. And the last thing I want to say is I'm really gratified that the FDA may be able to get its hands on patient-level data, because that's the only way you can really do this risk factor stratification. You really need to follow individuals. And don't forget social determinants of health.
Okay.
Harder to dichotomize, but just as important to stratify.
And let me just maybe make this more complicated, although I don't think it needs to be more complicated. But you said sort of where you can dichotomize things. But what about when you have individuals who have multiple neuropsychiatric symptoms, or have, how do you, obviously what we heard from the session before is what people want and need is they want to be able to find what is the appropriate therapy for the appropriate individual at the appropriate time in the appropriate manner, right? Which is a big ask. It's a big task. And that requires a lot of breaking down of data into these various pieces and parts, and no individual is exactly alike, right? I think we've heard people say that before, that, generalizing this sometimes doesn't help. So how do you see, and I'll throw this out, Paul, I'm not going to put Paul on the spot, but throw it out to sort of everyone. So how might the agency consider that scenario where risk modifiers may not be, this or that? It may be this, that, and, a combination of things. How might that be attacked?
Let me start, and I can be brief. You're really asking with symptoms, how do they cluster? And do you look at all the different symptoms, or do you cluster them, or do you just say any symptom counts? I think if you say any symptom counts, you're going to dilute any kind of a signal. If you have NPI, if you got neuropsychiatric inventory scores, which you very frequently do in these trials, you could simply start with the mild behavioral impairment nosology. That's an attempt. Mild MBI was really designed more for predicting dementia, but it's not a bad target. Other things people have done with the NPI is factored it into 4 factors. Actually, the psychosis factor is the most replicable of those. So yeah, there's different ways to cluster, but I think it's worth doing.
Would anybody else like to take a stab at this question? Valentina? Oh, you're on mute. There you
Yes.
go.
So this gets into, so these are questions that can be answered in the future because they pertain the way that clinical trials are designed. We already know that past, available clinical trials were not designed to distinguish. We can infer some of information, and we hope we will, to guide our, future designs. But that obviously gets into the clinical trial design that I don't want to be too specific about it. But obviously, what is happening is that as the quality of clinical trials improve, the quality of the measures improve as well as our scrutiny as an agency into these measures. So that if we have an indication, I'll just give you an example for Alzheimer's disease psychosis. We have Alzheimer's disease that's defined as an entity, and also we have psychosis defined at baseline as such. If we do know that psychosis co-occurs with agitation, which it does, we do try to ask the stratified randomization for the presence of agitation and try to balance across treatment arms that factor too, or at best monitor, during the study longitudinally as what happens with agitation so that we can, when we get the data set, understand how these 2 factors interplay. So that gets into the clinical trial design. That is not object of this workshop, as you've heard, but this is to say that this will be answered by future studies.
Okay. Would anybody else like to jump in there? I guess one of the questions I have, oh, go ahead, Chad.
I was just going to say quickly that, as a pharmacist, I'm always going to bring this back to the medication itself. And while I certainly recognize the need to pursue the different types of dementia and to get information as specific as we can to best treat patients, I also would look back to some of the data that we generated to create the warning that we've been talking about this afternoon, and it is not as specific. So I just would echo that we need to go back and really look at these medications individually. We need to look at their impact on mortality individually. We need to seek a mechanism for what might be causing the mortality risk. And we need to decide if each of these medications rises to the level of requiring that kind of a warning. Because ultimately, as specific as we can be about new medications and where they might fit, with a blanket warning, we challenge or we dissuade the use of those medications.
Yeah, and Chad, I think there was a question, maybe it was more of a comment that was submitted by the audience as well, that sort of wondered something similar that, I think the question was, why does the FDA include and expand I'm sorry, I'm going to flip into my black box warning. The box warnings to all antipsychotic class of drugs based on the analysis conducted 20 years ago that did not include the approved drugs in the analysis, even though the mechanism of action is unclear and are not well understood. So I think that was one of the questions. I don't know that we have I think we've kind of heard the answer a little bit from that's something that the FDA is looking to gather data on to be able to better tease out and answer.
And just a quick follow-up to that. It's just when we talk about mechanism of action, somebody brought up a new product with a different mechanism of action, but treating some psychosis. All of these medications really have different mechanisms of action, and what mechanism is the one that we're associating with the mortality risk. And I think that's part of the issue that we have on the front line side is, I know that these medications act differently. I know that we use these medications for different indications, but it doesn't seem that we evaluate the safety from that same specificity. We just blanket the medication class as being potentially unsafe. And I think that's part of the lack of transparency that we understand as clinicians from where this warning comes from.
Okay, Paul?
Dan Weintraub and I have been having a little side conversation I want to mention. Instead of necessarily saying you stratify by diagnosis, you might want to, where it's available, stratify by biomarkers. And that's because so many things overlap. So you might stratify by a positive p-tau2, plasma p-tau217. You might stratify by positive alpha-synuclein. It's particularly important for vascular, where our biomarkers are not as solid. So you might stratify by something about Physicus score on white matter hyperintensities, and you certainly might do a Framingham risk score or similar vascular risk. But think of it like that, rather than just trying to pin down the quote diagnosis. Pin down the biomarker instead.
I would agree with what Paul said. I think that the more that we can understand the underlying mechanism and pathophysiology, the more we can be feeding in more closely to a precision medicine approach and know which drugs and which approaches work for which individuals.
Okay, terrific. Thanks. I'm going to move to the second question that we had. And since we don't have it on the screen, I'll go ahead and read it out again. Can risk modifiers related to medical comorbidities be realistically accounted for in the analysis? What would be an adequate research method here? Rebecca?
So Nancy, I had some initial thoughts about this. I know there's many ways we could consider this through additional analyses, so in the retrospective data, but there is an opportunity potentially to start collecting some new information using real world data. Maybe something that we could even study using ALSNet, which is a real world data and evidence gathering platform that's following people today to inform both safety as well as efficacy of FDA approved treatments. And we have over 1,100 individuals already enrolled in ALSNet, and there are brexpiprazole data that are being collected from some patients. So that's an opportunity that either working with the FDA or even study sponsors moving forward, that we could really think about answering some of these questions, particularly in a more general real-world population where a lot of these medical comorbidities could be realistically accounted for, I think, in some of the analysis and the questions that we're asking.
Terrific. That sounds wonderful. Other comments folks would like to make? No? Okay. I'm going to move on to the third question, and partly we've got several related questions that the audience sent to this one, so I may sort of pull some of these together. But if a new product with an indication for neuropsychiatric symptoms of dementia is submitted to the FDA, how might the agency evaluate whether there is an associated increased risk in mortality, and if relevant, appropriately contextualize the risk? And that concept of contextualizing the risk has come up in quite a few folks' comments or questions that they posted online too. So maybe we can start more broadly, and then we'll dive down a little bit deeper into some of the questions that folks have submitted to the Q&A. Was anybody would like to take a stab at that? Chat?
Yeah, I think, initially at face value, that question is, how should the FDA evaluate safety? And I think the FDA should evaluate safety on the merits of the compound or the medication that's being applied for indication. I think you can't pull forward baggage from other medications or even within a class unless you can make the association of that new product to that class or that mechanism of action. And I think that's, again, sort of the core of the question here is these medications that may have been studied back in 2005 may be very different than the medications we may be seeing with application or recent application today? And how do we make the correlation that they are enough like those older ones to justify carrying that warning? I think, again, it comes back to evaluating on the merits of the medication that's being submitted. And in terms of contextualizing, I think that's an important point. You know, we need plain speak type of language for these warnings, even though we recognize these warnings are going to licensed clinicians and nurses and pharmacists and physicians. We need to, in a way that allows them to communicate it to their patients and to the caregivers. The way some of the black box warnings are written does not allow that simple communication.
Great. Matt?
Yeah, as a statistician, to me, one of the critical things to do this, and I'm not speak too much about the contextualization, that's a little more challenging, but is the trials are designed in a rigorous manner to collect mortality information, as well as any other serious adverse events that may be of interest to the compound. And really where that comes into play here, I think, is the trials are designed to continue to evaluate patients even after they discontinue treatment to understand the risks. We didn't get into this into detail in the presentation because it gets a little bit nuanced, but these on-treatment analyses have their own problems, of which we do have some concerns about from an analytical perspective. Whereas if you incorporate all deaths that occur up until, let's say, an end of double-blind treatment period, that is much more of an intention to treat type of analysis, which is somewhat more reliable from a statistical perspective and something we really do advocate for. So it's important that the trials do have rigorous things in place to continue to get vital status information on most every patient up until trial. And that's going to be so important to us to really understand this mortality signal.
Okay, great. Rebecca?
Thanks, Nancy. And I think just echoing certainly what Matt just stated, as well as what Paul had stated before about, or Chad had stated before about data. I think that's certainly going to be key. I think maybe more of an outstanding question more than a solution here, but getting to something that's related to contextualizing the risk. I think just moving forward in terms of the evolution of this reevaluation, it might be important for the FDA to consider how they might incorporate lived experience when making their final decisions about box warning and language as you're contextualizing the risk or characterizing the value of the treatment. I think we've all heard today that there's certainly a lot of emotion behind trying to deal with these challenging symptoms, but also people feel at a loss in what to do next. So I think maybe thinking about that in terms of contextualization might be important. How to actually analyze that in your decision-making, I think that's an open question. And I see Valentina shaking her head. That's going to be really difficult, but certainly encouraging incorporating this kind of opinion in your discussions will be important.
Okay, great. Paul?
It's a real problem, and I don't envy the FDA, because the duration and the N you need to find an efficacy signal is something that's practical to do, and that's why you've been able to approve a couple of drugs in the last couple of years. But it's probably not nearly enough to get a significant mortality signal. And so I encourage the FDA to encourage pharma to get all the data they can, including after the patient is finished, including open label data, observational data, death certificate data, because it's really hard to come up with those numbers. One reason why we've been doing these meta-analyses is that no one trial is powered enough to really see the mortality risk. I didn't solve the problem. I'm just saying that's the problem.
Thanks, Paul. Chad, go ahead.
Yeah, just to respond to Paul's comment, because I think it's a great comment. I think what we're faced with is the new product that comes with a new application is being asked to prove that they don't do something versus prove that they do something. And it's that assumption of sort of guilt from an old study that may be very disconnected from the product that's being evaluated that they have to overcome. And I think that's part of the transparency in the process that we need to understand from FDA is, why do you have to prove that you don't do something that we don't think that you would do?
Yeah, correct. Thanks, Chad. So this is, I think it's a somewhat related. Oh, Valentina, before I go to that, go ahead and want to comment.
Yes, I would like to respond to some of these questions. Like, for example, in the case of the Brexpiprazole review, there was an imbalance in that. We didn't apply the box because there was a box, there was a signal in there. So that's what I was the point I was trying to make. And then another point to Paul Rosenberg's observations, very correct. Sometimes, you know, we have a program that has maybe 2 studies, then we have another one and maybe has other 2 studies, and then we try to build our data sets on the basis of the in-house data. And that's necessary sometimes, you know, if we want to have that bigger picture. I wanted to talk about if we want to go in a direction. We don't know whether we're going to be revising the box as we go forward, but if we're going to go for a different regulatory action, certainly the way to go would be to pool you know, data that are, you know, obtained with the same data quality together, and obviously involve the public and the patients with the lived experience as we do in this type of decisions. But again, this is projected in the future. This is one way, one possible way we'll be proceeding. Let's put it this way.
Paul.
Anything we can do to help inform patients and families and clinicians of the magnitude of risk is really important, because it's crucial. The clinician is caught in between the family and patient saying, treat me, reduce my distress, and the regulators saying, there's a risk here. It's the magnitude of risk that's really important. As I said before, we're often in the realm of comfort care here, not necessarily palliative care, but a less aggressive approach. And in that context, families are my experience is that they're happy to accept some risk for a significant benefit. But it's nice if we can give them a number, something they can hang their hat on.
Well, and that's why I wanted to kind of get to touch a little bit. I know we're running short on time, but there was a question that came up in the Q&A that said, how can clinical trials assess how impactful any effects found in the trials are for patients and families so that the risk can be contextualized? So, I mean, is this kind of this is another element to this too, right? So, you know, trying to understand, and we heard this in the last session, you know, what is important, what is most value to the patients, their care partners, et cetera, and how much risk they're willing to take to have relief of some horrible symptoms that reduce their quality of life, for example. So is there a sort of a high level, I know we're running out of time, but kind of a high level thought on how we can use data from clinical trials to that pull information about what's important from patients, or combine things? So what we have from, you know, the Alzheimer's Association information and other things to pull together, what are the important things that we need to be considering when we're trying to contextualize risk here? Does anybody want to try to take a stab at that? Christina?
Sure. I think starting with the clinical trials, weaving in the measures that will capture caregiver distress and burden. I saw in some of the data that Dan Weintraub presented caregiver sleep and how that was impacted by a given drug. And so I think that if we include this kind of information as key secondary outcomes, you know, then we can track that and hopefully build a picture and data that will help contextualize this information.
Thanks, Christina. Matt?
Yeah, one of the things I would think that is extremely helpful in making a decision to take a treatment is if we would know the probability that I would, you know, die within, let's say, you know, 1 year of taking this treatment. I think that's extremely powerful and useful to a patient, to a caregiver. But that's where it's hard. You know, it's a relatively rare outcome, as people have commented, to really have really good ideas, you know, precise estimates of that probability. It requires large trials, and I don't think we probably want to go there yet. But that to me is the ideal situation. But I think we're going to have to probably work around maybe a less ideal situation to find ways to describe to patients what risks are, and to help the patient, and even the prescribers and caregivers are all going to be important. And I think it's going to be a challenging situation, but we can work on it.
Rebecca?
I think that we could maybe take some learnings too from what the Alzheimer's Association and many experts around the country have been trying to do in terms of contextualizing clinical meaningfulness, even of some of the newest approved treatments for Alzheimer's disease, and how can clinicians actually explain to patients what the benefits, but potentially the risk could be. So there's an opportunity to take some of those learnings as well in terms of what we can get out of the clinical trial data and how to interpret that. But also, I think some of those learnings could be brought into building and developing new clinical trials, as I think was posed by this question, and the types of data that we could capture, because we know what's going to be more meaningful in terms of helping clinicians describe the benefits as well as risks to patients.
Terrific. Okay, so in closing here, we got about 3 minutes. So I'm going to do kind of around the virtual room and give everyone about 30 seconds for a closing statement. I'm going to go ahead and go in the same order that we started. So we'll kick off with Paul.
I'm really glad the FDA is reconsidering its approach to the box warning, and I'm very glad that instead of blackening the box or adding a skull and crossbones, they are thinking of altering the language in a way that it'd be more useful. I thank them for thinking about that.
Thanks, Paul. Rebecca?
I think I'll reiterate that very grateful that we're having this conversation today, that it's definitely critical that there's options available for patients and families, but certainly that the options need to be backed by data to really support prescribing.
Great. Thanks, Rebecca. Chad?
Yeah, I would just reiterate that this has been a wonderful day of dialogue and discussion, and I think we're all on the right track to coming up with better ways to help ensure that patients are safe and that they also have access to the medications that may help them.
Okay, thanks, Chad. Christina?
Yeah, it's been a great day. This has been a very helpful discussion. I think we now are starting to develop some consensus about what are the steps that we need to do to move things forward, and we need to get to it.
Thanks, Christina. Valentina?
I'm very grateful for the important ideas that you gave us, important questions that you've raised, and suggestions that you've made. I would like to acknowledge the team, the FDA team that has worked in this project. It's a very large team of dedicated people, and I'm sure we'll all be able to take on your suggestions for the future.
And Matt?
Yeah, I just want to thank everyone again for participating, and I personally have really enjoyed the dialogue and getting to learn a lot more about this area. And I think, to me, where I end up is I think there's general alignment and agreement that it's important that we characterize the benefits and the risks moving forward and figuring out ways to do this. And I'm very optimistic through further engagement of sponsors, caregivers, our patient advocacy groups, that we're going to get there. So thank you.
Okay, great. And I want to thank all of you for your engagement and your active participation today. I think this was a great, insightful session and the beginning of the process. So we look forward to much more to come in this space. Now we're going to, I'm going to turn things over to Teresa Baraccio from the FDA for our closing remarks and to wrap up for the meeting today.
I'd like to express our thanks to our panelists and attendees for such an engaging event. I want to thank each of you for participating and for your thoughtful contributions to today's discussion. We are very fortunate to have such a distinguished group of experts with us today, and we value your perspectives. We heard a lot of great discussion. Thanks again to our presenters earlier in the day who provided an overview of foundational information to support conversation, including a clinical overview of behavioral and psychiatric symptoms of dementia, as well as relevant regulatory history of the boxed warning for antipsychotics. We also heard a report out of our recent meta-analysis of randomized controlled trial data, which found a higher mortality rate among patients treated with antipsychotics compared to those receiving a placebo. We heard findings from our review of relevant scientific literature. As highlighted during that talk, most observational studies seem to confirm, despite large variability in the data, a higher rate of mortality in individuals affected by dementia who were prescribed antipsychotics to treat neuropsychiatric symptoms. In session 3, we heard patient, care partner, and clinician perspectives on the value of antipsychotics for this patient population, and important context around how the boxed warning, as presented today, impacts patients and caregivers navigating neuropsychiatric symptoms of dementia. We know we have a number of outstanding questions to explore, and we heard some insightful thoughts, and considerations during the last session of the day. As other speakers have mentioned, today's conversation is only the first step in further characterizing the value and need for boxed warnings for antipsychotics for this patient population. There are several additional people I would like to thank for making this event possible. I would like to thank all the FDA staff who played an instrumental role in preparing materials and planning for this meeting. Many thanks to Valentina Mantua, Yuchen Zhao, Fengyu Zhao, Shamir Kalaria, Matt Sukup, Julia Birno, Jessica Jung, Paul Lee, Tiffany Farchione, Bernie Fisher, and Michelle Campbell. This meeting was truly a joint effort, and I'd also like to thank the Duke-Margolis team, Victoria Jem, Cameron Joyce, Nancy Allen Lapointe, Christina Silcox, Hannah Vidiello, and Luc Durocher, and for all of their efforts to put this event together. As a reminder, additional meeting materials will be posted to the Duke-Margolis website in the coming days, including a recording of this workshop. Thank you again for a wonderful event, and have a great afternoon.